Hello, 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 everybody. Welcome, welcome, welcome to the stream. Hello, I'm a little big right now. Let me go ahead and change that. Hello to all of you. It's been a while since I've done a legitimate journal club where I actually go through the paper before and do some slides. So I'm really, really excited about the topic that we have here today, because today we are going to be talking about something that affects many, many people worldwide, and that is IBS. But before that, I just wanted to chat a little bit while people file in. I know, at least on my coast, this is a pretty early time slot, but I, again, wanted to make sure that I can actually stream during Asia-friendly times too. Hello, Lithuanian Hussars. Welcome, welcome. Can't stay for the live stream. Sounds insane. I mean, I really do think it is. Hello, someone. Hello, Marlon. Welcome, welcome. Hello, Wilnot, Archie, A, Ilgaz. Welcome, welcome. Yeah, I wanted to just start off by sharing a thought that I had yesterday. If you're a part of my Discord, then this is something that you've probably already heard me say, but I had the realization that the number of biology students I have in my community is not insignificant, and at some point those biology students will become masters, they will become experts, and they will become better at biology than I am. And a sudden wave of terror overtook me as I imagined what it would be like to go from someone who people might look towards as a resource for biology know-how to potentially looking like a chump. Because I think once you start doing biology professionally, you might be able to see sort of where my faults and imperfections lie with regards to my expertise, which I think is a little terrifying. A little terrifying. All right. What's the stream about? This is about drug design. So I think this is maybe the first real drug design paper that I've covered on this channel so far. I don't really know if the average person has a decent understanding of how, like, why so much money gets poured into pharmacological discovery. It's actually a pretty interesting and mysterious process, I think, from the perspective of the audience. But today we're going to take a look at a very neat treatment that I think is neat in the sense that it is simple enough that anybody can understand. And I think those are the most elegant of designs. All right. Yeah. So, yeah, that idea of like, people are gonna look back when they become real professionals and view my stuff as kind of juvenile. But I mean, I got over it, of course. I think it's part of me thinks that like, oh, when they really know biology, like I don't even know. That's the thing. I don't even know what real professional working experts think of me, because why would they watch me? Although I do know that there are some biology graduate students in the chat and in my community, and the fact that they're still around maybe is an indication that I'm doing something good or entertaining or useful at a high level. But I don't know that for certain, and it's always a little spooky, I would say. Still studying bioinfo while watching stream. I mean, like at some point you will become an expert, and I really hope that when you look back on my content that it will have aged well. That's all I'm saying. All right. How do I answer the poll if I don't know if I have IBS? That's true. Many people don't actually know whether or not they have IBS because the symptoms are so non-specific. I think if you have a diagnosis of IBS, it must have been pretty severe. So that's actually maybe a pretty not useful poll to have to begin with, which I will talk more about right now as we get into talking about how new drugs are designed and discovered, which I've come to more recently appreciate how much I think this subject is awesome. Because if you've watched my channel for any appreciable amount of time, I think the best engineer out there is nature. And I actually did not think that I would enjoy working in an engineering field at all, because I thought, whatever nature is doing is probably more smart, more elegant, more beautiful than whatever humans can come up with. And at my recent job as a engineering instructor, I have learned that I was totally wrong. I think humans can be quite clever and fun too. And I hope this inspires some of you to maybe think more about engineering in a biological context, which I think is very neat. All right. Okay, so let's talk about this paper, shall we? Fishing out AIEC with FimH, capturing microgels for inflammatory bowel disease treatment. A lot of fancy jargony words. If you do, let's see if Stream Elements bot is awake right now. So let's do commands. Let's see, Stream Elements, because I have a glossary command that will, there we go, yeah. Have a glossary command that will actually enable you to follow along with all of the complicated acronyms that are not terribly complicated, but I will define these two right now. AIEC, those are adherent invasive E. coli. So this is an acronym for a specific type of no good evildoer E. coli, and FimH is a receptor on that E. coli. And this paper is going to attempt to treat IBS by essentially Katamariing all the E. coli out of, not all of the E. coli, all the bad E. coli out of your gut, which I think is really, really fun and really, really interesting. Okay. So I wanted to go over an agenda real quick, just to give you an idea of what we are going to be covering for today. We're going to talk a little bit about the principles of drug design. We're going to talk about target identification and mechanism, and a little bit about optimization and iteration, and then delivery. Right? These are all dimensions by which people who do drug design kind of think along. And then we're actually going to talk about this particular aspect, this particular example, which is designing a tool to treat IBS, which is a mechanistic menace. And then we'll get into the orb paper, as I call it. All right. So hypothetically, drug design is actually pretty simple, right? This is all drugs are, as simple as I can put it. There is some biological target. It's going to be DNA or RNA or protein or lipids, whatever thing is in the body. And most of drugs are just making a molecule that binds and changes that molecule in some productive way to make you feel better. And while this is a very simple idea in principle, in practice, this can be very, very challenging and very, very expensive. So I wanted to open up chat just by mining your preconceived notions about drug design and see how intuitive this process can really be. And so how would you discover a new drug? Especially if you are a non-biologist, I encourage you to try to come up with something, right? And even if it's very vague, even if it's very broad, I will still accept those answers because I think it's important to engage with these thought-provoking questions yourself before I reveal the answer. Do psychiatric conditions have any connection with IBS, considering how deeply linked they are in the digestive and nervous system? We will talk about that, Seed. The fact that IBS is not only something that we see effects of in the gut, but in the brain as well, which you can imagine anything that you loop the brain into is complicated, and IBS being a condition of the gut and having microbiomic factors, you basically just combine two of the body's most complex systems into a pretty nightmarish thing to have to explain. A says, look in nature, plant/microbes. Yeah. I mean, you can definitely look towards nature for inspiration. You can also test compounds found in nature. In fact, that's what most of pharmacology was for a very, very, very long time. Looks for drugs in nature, says James. Yeah. Nature is a great repository for things. But what happens when you run out of things to test for in nature? Not saying it's a good way, but maybe by mixing random herbs and chemicals and hoping for the best. I have no idea. That's not a bad idea either, as we will see very shortly. Have a bunch of rats and give them random chemicals and see what happens, / joking. DK, that's not entirely wrong for how people do it in the industry, believe it or not. It is very, very intuitive, right? You are all coming up with very intuitive ways to discover new drugs. And in fact, you are all correct, but you only have, I think, half the story here. So there are two main routes of drug discovery, and these are forward pharmacology and reverse pharmacology, or also known as rational drug design. And chat, your input is forward pharmacology. I think all of you are saying forward pharmacology. And Marlon's actually hinting at the reverse pharmacology side / rational drug design in terms of thinking about mechanisms. But we'll go first through something that I think is much more intuitive, something that most people have a general feeling might work, which is forward pharmacology. So forward pharmacology is the gotcha of drug design. So what do I mean by this? This is all the approach that you, chat, have told me. Right? So this is starting with a sparse or no hypothesis to a disease mechanism, right? We have a disease, we want to treat it, but we don't actually know enough about how it works. So we're just going to try some gotcha. So we'll throw hundreds, thousands, maybe millions of chemicals at a disease model and check for phenotypic improvement, meaning check to see if sick things get better. Now, this is not typically done in rats or mice or an actual living animal, because that is very difficult to scale up, right? You can't test millions of compounds on millions of mice. That's a speed run in order to make your company go broke. But instead of millions of mice, we could do hundreds of thousands of wells of cultured cells, right? So chat, your ideas are good. I'm just going to tune them a little bit for reality. So this is a neat approach because it is very intuitive. We just try a bunch of stuff and see if any of the sick cells, however we chose to make those cells sick, whether or not they get better, and we can worry about how they work later, right? So consulting nature is something like this, right? Like we can farm chemicals from nature. We can see what has historically worked from old remedies and old ancient medical manuals, and we just throw them on these cultured cells and see if they get better. So this is a very good approach. It requires no a priori knowledge of disease mechanisms. And so what types of diseases might this be a really good approach for? Like what kinds of diseases? If you had to say, like what diseases would do really well under this gotcha paradigm? While I catch up on chat. Modeling actual microbes. What do you mean by that, James? Like, what do you mean by modeling actual microbes? I think that's interesting. I think ethnomedicine is an important aspect in drug discovery. They have experience-based remedies, but we have the tools to filter which remedies work. Yeah, like traditional herbal medicine doesn't necessarily have to exist in a totally separate bucket than modern medicine. If traditional medicines work, they will be eventually confirmed and made a profit in the modern pharmacology world. I can't escape from the Genshin even here. Yeah, it's my go-to. Like, this is gotcha. I put Hu Tao here because I stopped Genshin after I failed to pull Hu Tao and burned everything I had on her. Gotcha is a very cool mistress. Leo says a new/unknown disease sounds like a prime candidate for this type of research. That's definitely a good approach for something that is relatively new. Golds says cancer. Yeah, I mean, cancer, we haven't fixed it yet, obviously. And so something that is very difficult to cure because we either don't know enough about it or it is so complex that a simple solution won't be sufficient. Definitely true. Maybe diseases with clear, easy to see symptoms. That's interesting. I'm not entirely sure whether symptomology is a factor for whether or not to choose forward or reverse pharmacology. Just because from the lab perspective, subtle diseases don't really matter because we can mimic subtle diseases in the lab. I think, I think maybe I misinterpreted what you were saying. You were saying maybe diseases are easy to recreate in the lab. And for that, I also agree, right? If you can make a cell sick somehow, right? Diseases are very complex, but if you have a reasonable way to make a cell look sick enough that reflects reality, that's also a prime candidate for this kind of thing. I assume infections would work well with this method, bacteria and parasites. Depending. You have to be kind of careful about how you approach this because the physiological context of a bacteria or a parasite is pretty specific. But if you can reasonably have this done in a dish, it's a good place to start, right? Obviously, this is not, you don't just like play the pharmacology gotcha and then immediately give that to a patient. You have to like test it in other contexts too, but certainly stuff that you can do in a dish would be a really good candidate for this. Okay, so the cool thing about this is that your target can be identified after, right? You don't have to know how your disease works in any great detail, but you can just see if cells get better, and then you can back solve for the target, which I think is very, very cool. All right, so I kind of think of forward pharmacology as being like unga bunga. Like it's clever, but it's not very elegant, right? You are throwing everything at the problem and hoping that you get a handful of good candidates, right? A screen of millions can just be can only like you might get garbage out of a screen for millions, like total utter garbage that doesn't go anywhere, and you're really hoping to find just a handful, like one, and you might be very happy, but often you find nothing, which is devastating. So there are pros and cons, as you can imagine, with forward pharmacology. One, you don't have to know everything. Two, you can test millions and millions of compounds, but it's you are really hoping for what a former professor that I worked with called pinches of magic dust, right? Like it is not a high yield kind of technique. Is this the monkey typewriter method? Pretty much. That's a very good way of putting it. It is the monkey typewriter method where you are just throwing everything at it. And you can imagine this is why drug design can be kind of expensive, or drug discovery can be kind of expensive, because this is not easy. It's not easy to find a pinch of magic dust when you can test literally millions of compounds and end up with garbage. Okay, the BOGO sort of drug design, I think so, but unlike BOGO sort, if I understand BOGO sort correctly, this is still a very good way to discover new drugs, because we have to acknowledge that we are not smart enough to understand every disease, right? This is something that patches up our limited understanding of how diseases work, right? If we had perfect information, a perfect understanding of every disease, you would never have to just try this, right? If we had perfect understanding of everything, we could just do the second approach, which is reverse pharmacology. This is the rational approach. And to me, this is like the sweaty gamer. This is the sweaty gamer method. This is the sweaty gamer method. I think this is a very, very intense, big brain, rationally designed. I think this is very elegant, and I think that, like, in order to make content, I'm probably never go, or maybe I would do like one forward pharmacology paper just to show you how it works. But to me, the more entertaining, the more interesting, the more intellectually stimulating version of drug discovery is this reverse pharmacology / rational approach. And indeed, students, when they try to design their own final projects, to do something in biological engineering, they will often suggest a screen, but we will often deny them, because just being like, oh, I'm just going to test millions of compounds to cure Alzheimer's. And it's like, okay, you could do that when you're a professional, but if we're trying to teach you how to think like a scientist, it is more valuable to learn a rational approach than the unbiased approach. All right. Okay. So, what does reverse pharmacology involve? So this requires knowledge of a target and a proposed mechanism, right? So this is something where you cannot do it for diseases you do not understand, right? If you don't understand anything, you can't possibly have a rational plan. Reverse pharmacology is a very fun for me mentally. Super cool to work through steps. I love problem solving. Yeah, exactly. Like this is fun. It can be emotionally devastating when your big brain beautiful plan doesn't actually work, but it is fun to read about, right? I don't envy these scientists who have to think rationally and then have their hopes dashed when biology is too complicated or doesn't work the way that they think it works. But again, that's why like millions, billions of dollars gets poured into developing these kinds of things, because scientists are more often wrong than they are right, and you just kind of have to eat that, which is an important part of being a scientist, right? We are professional people who are wrong all the time, but when it counts, on average, we get things right. So what this involves is, of course, designing a drug that is engineered specifically to attack said target, right? If you know what you're looking for, you know how to attack it, or you have an idea of how you want to attack it. So what diseases might be a good candidate for rational design? And so I kind of gave it away, diseases that we understand well, but like, do you know like the names of any diseases that this might be a good candidate for? And there are a lot that this fits under, to be honest. A lot of ones that are already out there, ones that are pretty prevalent in the public imagination, I would say. So any ideas of what you could do rationally? Any famous protein targets that are, something that you can really get a handle on? COVID is one, right? So Marlon, yeah. So we have the capsid structure of COVID, meaning we know what the virus looks like and we know how it attaches to cells, meaning that we can potentially design stuff that would block COVID-19 from entering cells. For example, right. Everyone's heard of the spike protein. So if we know what the spike protein looks like, and we very much do know what the spike protein looks like, someone could potentially try their hand at designing a drug rationally that attacks that spike protein. HIV, I mean, we know the mechanism well, but it's hard to develop for. I mean, HIV is getting its butt kicked right now, honestly. Like all the money spent on developing treatments and cures for HIV, I'm amazed to say to like understand that the world that we live in now is a very different world that we lived in the year 2000, where HIV is a very, very good candidate for rational drug design because of how much it is known about the disease. Although I don't actually know enough about HIV to tell you whether or not most of our current treatments that worked very well have been rationally designed or screened for. It's yeah, I'd say hypothetically it's a good candidate, but I couldn't tell you for certain if those treatments were rationally designed. Toxins, venoms, and poisons? Yeah, absolutely, because for a lot of toxins, venoms, and poisons, it's known what receptors they act upon. And so if you know what receptors they act upon, you can design something that blocks them. Yeah. Is this why there's a semi-conspiracy theory that the COVID vaccine was rushed? I think the explanation for COVID conspiracy theories will be much too complicated for a little guy like me to cover with any level of respect. Unfortunately, human says the cold. I don't know enough about the cold to tell you whether or not that is true. I'm not saying you're wrong. I'm personally not that well acquainted with how the cold works to confirm. All right. So let's go on. Oh yeah, runner-up candidates, I would say, like Alzheimer's. If you believe that beta amyloid is the candidate, then you can rationally design something against beta amyloid. That is, of course, if it really is beta amyloid, which people are kind of iffy about these days. Huntington's disease, which has a very clear molecular etiology, right? The Huntington protein aggregates in cells. That's almost for certain the thing that kills the cell. And so if you design something rationally against that protein or against the DNA or RNA that makes that protein, you can produce something very cool out of that. And actually, I forget who it was who posted the fact that there is now, I don't know if it was a drug in clinical trials or a drug that is out of clinical trials, but there is some recent breakthrough in Huntington's treatment that does appear to be rationally designed against the Huntington protein, which I think is very, very, very cool. Prions, says Leo. Yeah, absolutely. We know the prion protein. We know it aggregates. We know that it is sufficient to cause disease. I'm not saying it's easy to rationally design against prions, but you can do it, and I think that is very cool. And I think the current treatment in trials right now is also a rationally designed drug against the prion protein. So another point for rational design. All right. Okay, so even though these two methods of drug design start from different points, they do converge on the same endpoint, right? Which is the fact that it doesn't matter if you first have a molecular target or not, you eventually identify the target, you eventually identify how it works, and then you refine from there. This is generally how it works, although not always, right? You don't always have to know exactly how a drug works if it just does, right? So sometimes things can just skip the step of figuring out what it does and go straight to actually helping people as long as it's safe, right? And I think a lot of drugs in psychiatry kind of fit under this bill where, yeah, I mean, mental health stuff is incredibly complex. Like, it's kind of hubris to think that we could have a very, very deep mechanistic understanding of complex diseases that involve a person's personhood, right? So at some point you just have to be comfortable with, we don't really know how these drug works to a very high fidelity, but they work well enough and they have acceptable side effects, right? Of course, it's better the more we know about mental health, the more we will be able to rationally design drugs for conditions of mental health, and maybe not have so many side effects as we go on into the future. But I don't want people to be afraid of drugs that we don't have a clear understanding of, because sometimes those are the best tools that you have. Obviously, you would prefer if we knew more about what it did, but if it works and it's gone through safety trials, start here. God dang it. Okay. Hello, VOD watchers. I just made a huge mistake where I confused IBS and IBD for like the last hour. So I've cut out the introduction because that was all about IBS and not IBD. The things that IBS and IBD have in common are that they are both diseases of the gut with complex etiology. Not obvious how to tackle either one of them, but they are different. Whoopsies, my bad. No. I rushed too fast through making this presentation. Very, very embarrassing, but we will trudge on. Yeah, the principle still applies, but the specifics don't. So let's settle it there. Can't accuse you of being pre-recorded at least. Yeah, oh my god. Ain't no way this is pre-recorded. Okay. Oh, my face is so hot and I'm sweating because that's so embarrassing. But we will go. We will continue. Inflammatory bowel disease is an immune-mediated intestinal disorder with complex pathophysiological mechanisms involving genetic, environmental, microbial, and immunological factors. The rapidly increasing worldwide prevalence of IBD, driven by rising incidence and improved survival, necessitates urgent implementation of preventative measures and therapeutic advancements to mitigate its expanding public health impact. The primary types of IBD include ulcerative colitis, Crohn's disease, and indeterminate colitis. Unlike common intestinal inflammation, which is typically acute and curable with anti-infective treatment, IBD is characterized by a prolonged course with recurrent episodes and remains incurable to date. The clinical manifestations of IBD are varied, encompassing gastrointestinal symptoms such as diarrhea, abdominal pain, bloody stools, and perianal abscesses. Sounds pretty terrible. The etiology of IBD is multifactorial, including psychological, stress, autonomic nervous system dysfunction, dysbiosis of gut microbiota, and immune regulation associated with disease activity. Right, so yeah, IBS and IBD are similar enough where those general principles remain true, right? Psychological stress, autonomic nervous system dysfunction, dysbiosis, et cetera. But yeah, the specifics are not exactly the same. The primary characteristics of gut microbiota imbalance are an increase in mucosa-associated bacteria and a decrease in overall biodiversity. Although no single pathogenic microorganism has been definitively identified, many studies have reported that alterations in pathogens and commensal bacteria may be involved in the disease pathogenesis, right? So that's a pretty good introduction. I think it's very, very readable, which is great. I love reading nature papers because they are so friendly. People can argue about the quality, but I think it is not arguable that Nature is like one of the most readable journals out there. Okay, so then the authors move into respectfully acknowledging the scope of their treatment, right? Among these bacteria, adherent invasive E. coli is increasingly recognized as a major player in the pathogenesis of what CD is what? Colitis? What is this? CD, Crohn's disease. Sorry, Crohn's disease. Should have had an intro on Crohn's disease. Haha. Adherent invasive E. coli plays a crucial role in the pathogenesis of Crohn's disease through its ability to adhere to and invade intestinal cells, survive within macrophages, induce pro-inflammatory responses, disrupt the intestinal barrier, and contribute to gut dysbiosis. Upon entering the intestines, these E. coli strains adhere and invade epithelial cells via type 1 pili adhesin FimH, which has important clinical significance in the pathogenesis of Crohn's disease. Specifically, FimH directly binds to mannose residues on carcinoembryonic antigen-related cell adhesion molecule 6, or CCAM6, glycoproteins present on the surface of intestinal epithelial cells, which facilitates the initial attachment of E. coli to the host cells. Okay. So this is basically saying we don't really know how Crohn's disease works, but we do suspect that this bad E. coli is responsible for a good chunk of it. And so we know the targets that we're going after because we know that this bad E. coli likes to bind host cells via its mannose receptor, which they call FimH, right? This type 1 pili adhesin FimH. So the system is E. coli. Let me just draw. Ooh, do I have my drawing tool handy? Docs. Do I have draw? Where's my drawing tool? No. I updated OBS and I don't know where my stuff is. Give me a second. Oh, I would love to draw on this. That's so lame. No, I should have tested the doc. Sorry, give me 1 second to try and find. No, I just don't see the drawing doc anymore. That sucks. Okay, so try to imagine this in your head. E. coli, bad E. coli, they invade cells. They enter cells and invade cells, right? They are intracellular to invade them, right? And so in order to invade the cells, they need a point of attachment. And the point of attachment between E. coli and your gut cells is this mannose sugar that is on your host cell. E. coli binds that host sugar with its FimH protein. So those are the players here. And again, thank you, Celeste and Marlon, for bringing up the glossary, reminding people that they can actually bring up these acronyms anytime they want to in this story. All right. It's interesting to think of sugars as structural components of biological tissue, even outside of the context of cellulose and plants. Yeah, sugars are really, really important for biological signaling. So you might think, why does this cell have mannose on its surface to begin with? If it enables E. coli to invade the cell, it's because the trade-off is worth it, because mannose sugars on the outside are an important way for cells to communicate with each other. And also, like, they're important parts of biological membranes as well, making sure that they are correctly structured. All right. Okay, so then they go ahead and they identify the gap here, right? They identify what they are planning to do about the problem. The treatment of inflammatory bowel disease still faces significant challenges as most current targeted strategies exhibit limited therapeutic efficacy and are often associated with adverse effects and the emergence of drug resistance. Recent studies have shown that disrupting the crucial interactions between CEACAM6, FimH, and mannose can effectively reduce the pathogenicity of bad E. coli, offering a promising approach for the management of Crohn's disease. However, these therapeutics often suffer from poor target selectivity, limited bioavailability, or the need for repeated administration, and their stability and safety in the complex intestinal environment remain insufficient. All right, so this is not the first paper to try to disrupt E. coli binding to FimH, right? This is not the first attempt at this. And now, like, I think the most recent sort of trend seems kind of rude to put it, but like the recent push in drug design, I think, is all about delivery because, yeah, I think it's like we know enough about biology in a lot of contexts to figure out what we should do, but not necessarily know how to do it. And so this paper is taking this concept that we can break E. coli's attachment to your intestinal lining with something, but those somethings so far have not been great. So here is the gap that they are pointing out, and we're going to see whether or not they actually fill these gaps. So poor selectivity, right? They don't only do the thing that they purport to do. Limited bioavailability, so they just don't work very well. Repeated administration, like ideally you want to develop a drug that works one time and then you're good forever. Although if you're a pharmaceutical company, maybe you don't really want to invest in that kind of thing, but we're talking about the level of basic research, right? Any gripes you have with big pharma, you can take to the companies. At the research level, you know, there are still treatments out there that we want to be ideal for the patient. And then stability and safety, right? Safety you can't really get too much into in an exploratory paper, but yeah. So we'll figure out whether or not those gaps have been, whether or not those gaps will be improved. Also, just the thing on the pharma skepticism, right? Do I think pharma has a financial incentive to keep people on drugs for a long time? Yeah, but I also think that they exist in a very competitive landscape where if one pharma company decides to make a great drug that is like uncontroversially good, they can get a competitive edge, right? So I think like there's a conspiracy theory that big pharma are suppressing cancer research. I don't think that that's true because the second that one pharma company figures out how to cure cancer, you don't think they're going to make a gajillion dollars off of it and other pharma companies are going to try to compete. Yeah, I don't think these blood-sucking, thirsty pharma companies are gonna be outdone by any of their neighbors with regards to like big things. But I will acknowledge that like in terms of whether or not they choose to invest in certain treatments might be informed by their bottom line. So I think it is complex, right? I don't want to be too critical of big pharma, but I also don't want to let them get off scot-free, right? Like my channel is all about skeptical balance, right? So be vigilant, but also I don't think pharma is like mega evil at the same time, right? It's capitalism, baby. And that's all I'll say about that. Okay, so then they propose their treatment, right? Microgels, micron scale granular hydrogels have gained significant attention due to their small size, high flexibility, good injectability, porosity, and large specific surface area. Such unique characteristics make them a promising option for use as carriers in drug delivery and cell therapy. However, traditional microfluidic fabrication often involves oils and surfactants, limiting biocompatibility. The ATPS-based strategy offers an oil-free, surfactant-free alternative, enabling the biofabrication of microgels for applications in tissue engineering, drug screening, and disease modeling. Right? So this is basically saying if we, our previous methods involve things that are less friendly to a biological environment, including oils, surfactants, et cetera, and we have an ability to make these gels without those troublesome compounds. I won't call them harmful, but maybe not optimal, right? So we'll learn about what this ATPS strategy is in just a little bit. At least we have things like HPV vaccine that prevent cervical cancer by a lot. Yeah, exactly. Like people who go, oh, big pharma are suppressing cancer treatments. Like big pharma had the opportunity to suppress a whole lot like vaccines, right? Like vaccines are so good, right? And if big pharma wanted to pump out drugs forever and keep you all hooked on medications for life, they wouldn't be interested in making any vaccine, right? And the fact that we have great vaccines out there means that there is balance, right? The competition of pharma is balanced by their incentive to do profit motive stuff, right? So it's complicated, I'll say. But yeah, that's an excellent example, right? Like, well, yeah, we could just treat HPV forever and put you on meds for life, or you could have the HPV vaccine, which is proof that pharma can still do things that aren't polluted by profit motive. Yeah, I mean, I'm a biology content creator, right? Like I kind of have to be pro pharma in a way. Turns out that scientists working together to make drugs to cure diseases is like a good thing, right? It is where capitalism gets involved where the discussion gets a lot trickier. Okay, so here is where they show what they will do and give you a preview of what their strategy is. Here, as schematically briefed in Figure 1, we present a robust method for fabricating porous microgels loaded with oligosaccharides, MOS, and oligomer of mannose. So an oligomer is just, you can think of an oligomer as just multiple chains of certain something. So an oligomer of mannose is just mannose bound to mannose, bound to mannose, bound to mannose, right? These are chains of mannose. By merging all aqueous and coaxial microfluidic technologies, instead of directly delivering drugs which may lead to toxicity or resistance, we propose a competitive adsorption strategy where microgels, by leveraging the specific binding affinity of FimH to mannose, selectively captures bad E. coli and reduce its colonization of intestinal epithelial cells. By lowering the abundance of these bad bacteria while sparing beneficial microbes, this approach may help restore gut microbiota balance and potentially alleviate inflammatory bowel disease. All right. So this is the picture that we have here, right? So on here, oh man, I really wish my drawing tool was here. That's so annoying. Whoa, where is my drawing thing? Please. That's so sad. Dang it. Anyway, the figure over here on the left, let's see if I can get my mouse to show up on OBS. Yeah, there we go. So this is a zoom in of the membrane of the epithelial intestinal cell, right? Here we have the membrane, and here we have this membrane sugar, right? There's a protein receptor, and at the tip you have the mannose sugar. Turns out E. coli is really, really sticky for this mannose sugar. As you can see, all of these E. coli want to bind this mannose sugar to put them in close enough proximity to your intestinal cells to actually invade them. In order to deal with this overgrowth of bad E. coli, the authors present this microgel, this bead, this orb, this sphere that can compete with E. coli, that can compete with the host mannose to rip E. coli off of your intestinal cells, preventing them from invading your cells. And I think that's cool. It's cool, and I can explain it using plain clothes language, right? I can explain it using language that doesn't have to be very fancy, and those are my favorite types of drugs to describe. Okay. So I liken this to Katamari, right? I don't know if any of the authors here have ever played Katamari, but I think of this as Katamari, right? You have just things that are in the environment and you roll them up in a big ball, a big sticky ball, and then you roll it out of there, right? So I think this is such a fun idea, and I think it's something that anybody in chat could have come up with if they thought long and hard about. The fact that no one else has tried it like this is what makes it so brilliant, right? Like this explanation of, yeah, we're just gonna make like little sticky balls and roll E. coli out of there. It's like, damn, that's such a easy, simple idea. I wish I thought of it. Like, that's not to take away from the brilliance of the scientists, right? It's often these very intuitive, very elegant solutions that require a level of insight and brilliance in a scientist that, because if a scientist comes up with a complicated thing that takes forever to learn about, I'm not that impressed, honestly. I mean, it could be very impressive in terms of what it can do, but in terms of the feeling of like, oh, that guy's really smart, like, I think this is really smart because it is simple, and that is like a me thing, right? I think that is true. Ad meaning away from, so it competes with the gut lining to absorb the E. coli cell. Exactly. Isn't competitive absorption an artificial competitor to E. coli? Yeah. So they are. So E. coli is binding the mannose. I mean, I think you can word it either way, right? Competition is basically you're taking the beads and you are peeling the E. coli away because the E. coli would rather bind the beads than they would bind the mannose sugar. So I'm not too picky about the way you word it, but the effect is the same, right? The bead is competing and tearing away E. coli. Is the cleverness coming from how to design a sticky ball? I think the cleverness is thinking of this to begin with, like just thinking of the sticky ball, and then where the hard work comes in is, okay, well, how do we make that sticky ball, right? Because sticky ball with nanos on the ends, that's pretty easy to think of. The actual thing that you need engineering expertise for is understanding how to do this, which they are using this ATPS strategy, which we'll get into right here. So what is ATPS? Why do these scientists have like expertise, and why were they able to do it and the average person not able to do it? Well, it involves these very specific manufacturing techniques. This is a technique that I am not very familiar with. This is the first time that I've seen it. This is called an aqueous two-phase system. So if I explained something wrong here, I'm sorry. This is my best understanding of how this works. So the way that this works is that you are smashing two immiscible liquids together. So immiscible, if you remember from chemistry, is means non-mixing, right? Two liquids that do not play friendly with one another. For whatever reason, they don't mix. Immiscible, like oil and water are immiscible, but oil is like a fat. So I don't really know if they would count as aqueous, right? It is not quite the same. But two liquids that don't like mixing with one another, and you put them in this microfluidic device where you pump one stream of the aqueous phase through this narrow tube, and then you fill the chamber with another thing, and then it gets tighter and tighter and tighter until eventually, like, the pressure from one immiscible liquid will pinch off little beads. And then once these little beads get pinched off into, and don't worry, there'll be another picture to see how this works. But once you get that pressure, you form these beads, which you can then use UV to cross-link. So it's kind of like instantly making sure that these water beads become solids. And you can kind of see here that you get the mixture of these two immiscible fluids. So this is a cheap and fast way to make a biocompatible thing that can hold things, right? Because a lot of drug delivery stuff involves surfactants and oils and things that can make it a little hard for the body to actually use. And this is two liquids, and two liquids are going to be very, very easy to do something with, as opposed to something with a membrane or oil or other chemical. Am I wrong? But with competitive in biology, like competitive binding with enzymes means that the number of bioavailability, the number or bioavailability is the defining factor. I don't really know what you mean by that defining factor. You might need to define defining factor for me. Okay, so yeah, once the microfluidic device gets thin enough, you start getting these beads that get pushed out, and then you can use UV to cross-link one of the ingredients to form these polymer nets that holds everything together. You can think of it like gelatin. In fact, I think this is gelatin, or I think this at least contains gelatin. So these beads are dressed in mannose, the same thing pathogenic E. coli sticks to on your epithelial cells via its FimH receptor, right? So these beads, you can see, they have on the surface, or I'm actually not sure if it's on the surface or throughout, but they are embedded with the nanos, which I think is very, very cool. Defining factor was a misspeak, a very important factor in what the object will bind to. Yeah, so yeah, I'm still not entirely sure how to address that comment, Celeste. And let me reread the comment. Am I wrong with competitive biology? Like competitive binding with enzymes means that the number of bioavailability is defining factor. So you can think of like there being a certain number of binding sites on the intestinal epithelium, and you can think of them as like E. coli like hugging onto it. And then when you flush this bead through, you can actually, like, yeah, it's exactly just like the Katamari thing. You just roll them over And like Velcro or something, they pick up the E. coli, you can flush them out. Okay, so this is one of those glossary terms that is going to come up. The MOS MGs, this stands for mannose oligomer microbead. Right, yeah, oligomer microgel. Sorry, this is microgel. This is not microbead. So when you see MOS MG, just replace that, Control F that in your head and replace it with a little bead treatment. They look like beads, so I call them beads. But they're called mannose oligomer microgels. Okay, so that's how that works. And I will pause here for any questions that chat might have with regards to how this technology is supposedly going to work, right? Supposedly going to work. Any questions on this, right? The little Katamari ball that they spit out, and this Katamari ball is sticky specifically for E. coli. That is the most summarized I think I can do that. That is the most summarized. Yeah. Any questions before I move on? I think this is very cool though. Like once we get into a zoom in on this picture, you can actually kind of see these beads form in real time, and like to me that process is very elegant. Like giving props to humanity, right? Humanity did a good thing. Was mannose one of the aqueous phase liquids used? So mannose wouldn't be a liquid. Mannose would be dissolved in that liquid, right? You can see here, I believe in this phase that's on the outside, this is where I think this is mannose up here. This is where that mannose sugar lives. So you can see that these beads are going to be coated with the phase that has the mannose in it. So it's not mannose. Mannose isn't the phase. Mannose is in that phase. Oh no, did I program the MOS MG? I think I programmed that wrong, Marlon. If it's not coming up for you, let me see if I spelled it correctly in the glossary. MOS MGs. Ah, that's my bad. Let me fix that. Let me fix that command. Because I set up the stream element spot wrong. So let's do that. Okay, so that should work now. If you do exclamation mark glossary, that should give you the correct spelling of what the term should be. My bad. Yeah. Do you immerse them in beads again in the inner liquid to avoid them from mixing together like tiny cells? There's a complicated wash process that I skipped over because I didn't feel like it was interesting. But if you look at the paper, you can find details on how they cleaned up these beads. In order for them to stop mixing again, however, that's this cross-linking step. So over here in this corner, you can see that once you they'll shoot these little beads out into a dish, and then on the way there, they will be shot with UV light, which cross-links the polymer within the bead to make a gelatin-like ball. It's actually gelatin and not something indigestible by pepsins. It's gelatin and this cross-linking reagent LAP. So a gelatin is a part of it, I think, to give it structure, but then the cross-linking comes from something else. And because the outside is this mannose phase, I think that keeps them not removable. Or like, I think you excrete this. Although I could be wrong on that. It might be case sensitive. Sorry. Wait. Wait. Yeah. Sorry. It should be this. MOS MG. Stream elements bot, please. Okay. There we go. Yeah. Okay. Yeah. I updated the glossary and not the actual thing. Yeah. Okay. So. Let's see. Is this technology theoretically going to improve these things? Theoretically, mannose binding bacteria are generally the kinds that like to invade your cells, right? Because your regular healthy commensal bacteria, not all of them make a mannose binding thing, because why would they? The commensal bacteria that you have in your gut lives primarily on top of the mucin layer. So your cells are coated in a layer of mucus, and the cells that like eat, like they can be floating around in the intestine, they can be sticking to the mucus and eating the sugar that the mucus contains. they're generally pretty chill guys. It's only the cells that want to do you harm that will tunnel through that mucus and try to invade your cells. So, and you know, this might not be true for all bacteria, but there are quite a few bacteria that have mannose receptors that like to invade and hurt you. So I think this is a pretty good theoretical improvement, right? Mannose binding, if it is shared amongst bad bacteria and not amongst good bacteria, it could be a pretty good way to discriminate between bacteria that are good for you, bacteria that are bad for you. And why might this be better than antibiotics, right? Because you could argue, well, if IBD or IBS is caused by bad bacteria, why not just nuke the gut biome with an antibiotic, right? Why might this be better than just treatment with antibiotics? And you know, okay, besides the fact that IBD doesn't have a single bacterial origin, just like hypothetically, why might this Katamari strategy be better than an antibiotic? Only targets the mannose binding bacteria instead of all of them. Yeah, because you know, antibiotics are not exactly very specific, right? A lot of them have to do with just breaking bacterial membranes, which, you know, a lot of bacteria have membranes and a lot of them have membranes that are similar to one another. So if you are nuking the microbiome, you're going to be experiencing some unpleasant side effects. And so if you are just Katamariing the bad bacteria away, that's hypothetically a lot cleaner, a lot more specific. And it just goes to show, like, when I was a kid, I was like, dude, science is so goated, right? We have antibiotics, we have antivirals. Surely science cannot get better. But then, like, improvements in specificity, reduction of side effects, these are all, you know, very active areas of research, specifically because we understand biology more today than we did years ago. Yeah, fewer side effects, Thomas. Exactly. And, you know, as Piffy says, deter antibiotic resistance. There's part of that being true, right? I think there's an interesting question as to whether or not you still can develop resistance to this because you are technically removing the bacteria from their location. While that might not be literally killing them, I still think it puts pressure on the bacteria in the intestine to evolve a way to run away from the Katamari, essentially. I don't know if that's necessarily true, because this is kind of a neat thing where, like, they're not really dying, but they are being prevented from reproducing in this particular place, which might result in them dying later, if that kind of makes sense. So I think for certain it reduces antibiotic resistance, because this is by definition not an antibiotic. But I wonder if you could develop resistance to this too. It's pretty interesting, I think. Okay, and hypothetically, limited bioavailability is a problem. I don't know a lot about ATPS system, but they say that it's more bioavailable because it's 2 liquid phases. Sure, you know, I'm not an engineer in this particular field, so I don't know. I guess I agree with that. So let's take a look at how this actual thing works. So you can see that, like, a lot of engineering is done very, very simply, right? This is just a little UV light, and then this is a little microfluidic device that you can have on your benchtop. And then you have a very, very thin tube of your components, right? So you have the continuous phase that's on the outside, and then you have the shear phase, which is in the middle. So you have this device that can shoot into different liquids, and eventually the tube gets so small that you start getting pressure from the continuous aqueous phase that will pinch off these little beads, which are really, really cool. So this is a fluorescent example where it's just the filament. And then in the second panel, I believe this is where you start seeing the beads actually get pinched off. So as the pressure from the walls increases, I believe that is cinching off these little beads, which is so cool to see. Like, it's a very, like, again, when I was an undergraduate, I did not give engineers enough credit because I thought nature was so clever. But this is really clever on its own, right? Like, you make that small tube, and that small tube puts pressure on the surrounding fluid to pinch off the inner fluid. And because they are immiscible, because they don't mix, they form these little beads, which is really, really neat, right? And then you can get a close-up into what these beads actually look like. You can see these matrices, the texture of these microbeads. So cool. Can this fabrication technique be extended to produce a more general category of therapies? Yeah, you can think of this as a, I don't think they call it a drug delivery system, but a, because I think before this paper, from what I briefly looked over an ATPS review paper just so that I could like figure out what was going on, and this can be very useful for like protein purification and other lab techniques. Like this has, I think it's a relatively recent technique that people thought of that you can imagine could have all sorts of different applications. I don't know whether or not an antibiotic would be appropriate for this method of delivery, but that is not because I have a good reason. That is just because I know so little about this ATPS system. Little did he know engineers take inspiration from nature. I know, but sometimes when engineers take inspiration from nature, it feels like, it's like you got that from nature. But I feel like this is taking inspiration from like chemistry, right? This is like phase chemistry, liquid phase chemistry, which I think is, it's not like copying nature's homework. This is like, oh, we understood, like we didn't look at an animal and figure this out. We did chemistry for a long enough time to be able to apply it to biology. And I think there's something quite intriguing about that. Not that like nature inspiration isn't good. I do think that that is still pretty cool. But to me, that is more about the nature than it is about the scientist. Whereas this is applying phase chemistry, which I think is really, really novel and ingenious. Very, very neat. Very, very cool. Love that. And this is coming from someone who has a chemistry phobia, right? Like if you watch my neutrophil video, I'm not great at chemistry, but like something like this, like I think I can still appreciate. Yeah, you just shoot these little beads in these little dishes and you can even see them, right? You can see these little beads form. They are visible to the eye. Very, very, very neat. I feel like this can be used in tissue cultures, like making synthetic pancreas islets. Yeah, maybe. I mean, correct me if I'm wrong, Thomas, but you are in an engineering field, right? Like, I don't know. Do you know about this technology, or is this new to you? Okay, so are these biobeads biocompatible? To which I will interpret as safe, right? Because I think biocompatibility is a little of a broad word, but I'm going to interpret it, and maybe I'm interpreting it wrong, but I'm interpreting it as safety. So here's some more thought-provoking things for chat. So here I have a cell death assay. So PI, propydium iodide, is a cell death marker. So this is a fluorescent dye, and this dye should only show up in dead cells, because if you are a cell that is alive, you will be pumping out the propydium iodide, right? So if you see red, that means the dye filled the cell. That means the cell is dead because it can't get rid of this dye. Calcium AM, which should show up as green. This is a dye that is cleaved inside the cell. So this is something that is colorless unless it is inside a living cell, because the living cell has an enzyme that breaks this dye such that it gains fluorescent properties. So presence of red is dead, presence of green is alive. And so the cell type here is immortalized human colorectal adenocarcinoma cells. Say that 5 times fast. And I don't know what concentration is shown here, but in this experiment, they did a combination of an increasing concentration between 0 to 20% weight by volume of these microgels. So they are really cramming these microgels full. And I want chat to just kind of look at this image, knowing what you know, and tell me whether or not these beads appear to be safe. I'm in biotech taking a tissue engineering elective. Oh, very, very cool. So you probably will be, again, speaking of like students becoming masters, like I'm very much an amateur in this field, right? Like I've never taken a formal engineering class. I got hired based on my biology expertise. So I'm excited to see like how you, like, maybe you read about ATPS and never think about it again, but like, it'd be cool if you end up using ATPS or see ATPS come up more often in your burgeoning career in biotech. That's so cool. I can't see any red, and I'm definitely not colorblind. That is true. So I would say that I also can't see any red. So what might that suggest? Are these beads safe to use? So I learned a little of these techniques like microfluidics. Yeah, I had to pick it up on the fly. I never had a microfluidics class, although it would have been really cool. And because I never had a microfluidics class, when I talk about microfluidics, I'm always a little nervous. I'm gonna get called out for not knowing anything. Well, we see no PI and we see a lot of green. Yeah, so what might that mean? Interpret that. That's the observation. I'm very glad you started with the observation, Marlon, because you should always start off by just saying what you see. So like Seed saying, I don't see any red. Marlon saying we see no red, but a lot of green. Now you can interpret. So what might this mean? So given what we know, it looks like this evidence suggests cell survival. And I would agree with you. I would agree with you. However, if we turn up the brightness, what do we see? So I took this image and it looked a little fishy to me. So I went into Photoshop and I cranked up the brightness, and now what do we see? Now what do we see? So they don't kill cells. That is what I would say. I think it looks pretty good. Based on this, it probably means it's safe, doesn't kill cells. I like what Leo did there too, because there's the probably it's safe, but then quantifying it with doesn't kill cells, because doesn't kill cells doesn't always mean safe. So thank you, Leo, for making that a distinction, right? Because you don't want to be lulled into a false sense of security. It doesn't kill these particular cells, so it might be safe. That is the appropriate thing. Yeah, when I punch up the brightness, we do see some subtle redness. Do we have a scale? What level of each is significant? There's not a whole lot of detail, which is kind of annoying here, right? But we do see when you increase the brightness that there is a bit of red, which, you know, this is not something to be that concerned about, but it is worth noting, right? So this is not like, oh, these people are like fabricating data, you know. I never assume that, right? I never assume the worst. I try to always come up with a way that this could be explained without, you know, nefariousness at foot, right? So I tried to look at their methods for evaluating the toxicity of these beads. Samples were placed on round glass slides at the bottom of a 12-well plate. These cells in HeLa were seated in the plate. Live dead staining was done on day 1, 4, and 7. Images were captured using fluorescence microscopy with excitation at these wavelengths. This is all well and good. Green fluorescence indicated live and red indicated dead, allowing for assessment of cell number and viability. This is super annoying because I don't know what this means, right? Red fluorescence indicated dead cells. Like, what are we doing? Like total, like red fluorescence as a number divided by like live. Like, what are we doing here? Are we doing a threshold? Like, if there's enough red in a cell, we count that alive. If there's not enough red in a cell, we count that as dead. Like, there's just not like a lot of detail as to how they did their analysis. And again, like without the and to be clear, this green looks great. This is how it should be, right? This brightness is great. This you should not publish because this is losing data because you cranked up the saturation, which means there's pixel information loss. So you should not do this to all of your images. But the red, I don't know how they did their fluorescence microscopy. I don't know how they did their analysis, but it does look like there is more red on day 4 than there is on day 7 or day 1 for this set of images that they chose to show. Now, you know, if I wanted to be very, very mean, I could say maybe this indicates that this is not very reliable data, but I also want to point out that this kind of thing, you know, you would only know if you did microscopy yourself, but sometimes some slides just have a higher background, right? I'm not like super concerned about this, but I usually keep note of this because, you know, this is what scientists do. You're picky, right? The red might not be inside cells. Like maybe some of these redness that shows up in here is just like stuck on the glass, but unfortunately we don't have a nuclear marker or cell membrane marker to be really sure. So I don't know how they figured out which ones are dead, which ones are alive, which I'm kind of annoyed by. I'm inclined to believe the authors here, but yeah, the PI in day 4 seems a little weirdly red to me. Like maybe normalization to some negative or some positive control would have helped. Like, I don't know, some thing to clean up how this looks. You know, and maybe other microscopists think that this is not a big deal. Like if it looks like this, maybe it's not a big deal. I think it's just like a tiny bit suspicious. It's not like a major thing. And you know, it's been a while since I've personally done microscopy, but like to me, like, yeah, I know I had to increase the brightness here, but it does look more, and that's a little weird, but not enough for me to care, right? Like, this is me avoiding research hell, right? Like when you're going through a paper, you're reading it for Journal Club, you are at your maximal skepticism. And here I'm doing skepticism, right? I'm analyzing the data, I'm forming my own opinions, but not every imperfection is like death for a paper, right? I think even though you can see the red, it's not that high. And, I'm a little annoyed that the methods aren't very complete, so I can know what went on here. But again, I'm inclined to believe them. Although I really do wish they explained a little more about specifics on how they counted dead and alive cells. Yeah. So I don't know how they crunched their data. I don't know, what their methods were. And because live dead is quite common and old, many people kind of skip explaining how they do it, which again, to me, I don't think you should do that. I think you should still explain how you counted live and how you counted dead. But maybe that's just me. Maybe I'm old fashioned. But once they crunch their data, it doesn't look like day 4 is all that much different than day 1. Which again, maybe it's a little fishy, maybe it's not. But, that's something I usually keep in mind when I'm interpreting the rest of the paper. Like, if this is the only bad thing about the paper, I'm probably going to forget that this even happened. But like, if there are multiple iffy things in the paper, that's when everything starts to look a lot worse. And this is how you can engage critically with the paper without being a party kill, right? Like, I don't mind telling the person who wrote this paper, your day 4 looks a little high background. They would probably agree. But if I were to tell them that means this is garbage, they would probably disagree. Right? I'm trying to model responsible scientific engagement. Yeah. It's always the dang brightness. Yeah, but again, like this is fine, right? This image is fine. It's just that I noticed that the background in the middle was a little high, so I wanted to see how bad it actually was. Because these green ones, if they're this high, you should not do this. This is bad. Okay, so once they did their numbers, however they did it, and again, I'm a little miffed that they don't explain exactly what they did to count the cells, but maybe that's just me being grandpa. How much are you convinced that these microbeads have the potential to be safe, right? And Leo kind of teased this before, but once you see this data, on the left here, we have the control where there are no microbeads, and then all the way to the right, we have 20% weight by volume microbeads. So they cram these dishes filled with these microbeads. And what can you tell me about your interpretation on their safety profile? Leo kind of had an answer before, but if you're new to the stream, here's your chance to have another crack at this. Having worked in cell culture, cells will die from literally nothing. Yeah, and sometimes your like glass slide is just a little dirty or sticky, and maybe you didn't wash that slide well enough. And it's actually nice that they didn't cherry pick a better looking image because, in papers, you have to pick representative data. You don't just put every picture you took. That's just something that's supposed to be representative of everything. So they could have chosen something that looked a lot better for that second picture, but maybe all of their day 4s were a little funny. Maybe on day 4, they just like washed it a little bad and all of their images had a little bit of a higher background. But that's even more important as to why I think they should have included their analysis methods. Just for picky people like me, I think. Potential to be safe, sure, but it maybe causes harm in some method not captured here. Yeah, I think that's a perfect encapsulation, right? It doesn't kill these cells, these particular colorectal adenocarcinoma cells, right? I think it's okay. I think the safety profile at this stage for this type of paper where it's just making the stupid thing and testing the stupid thing, this is fine by me, right? There's no obvious toxicity in this cell line. However, you should note that these are immortalized cell lines, so they're pretty hardy, right? As Gold says, if you work in cell culture, a lot of the times they die. Immortalized cell lines are like pretty tough, right? Anything that comes from something cancer, cancer has been changed to professionally survive, right? Whereas your non-immortalized cells have things like apoptosis to keep them in check, whereas cancer lines are incredibly hardy. So I wouldn't take this to be, oh, they're completely safe, but as much of you in chat are expressing, it's safe enough to continue to explore, right? If these things poisoned these immortal cell lines, okay, maybe you don't continue this line of research, because if they poison immortal cell lines, think of what they could do to your other cells. So 20% weight by volume seems very, very well tolerated, even if I think the sample image is just a tiny bit sus. And it's normal for a paper to have things that are a tiny bit sus because science is imperfect, but you should still keep in mind all the good things and all the tiny bit sus things. And then at the end of the day, if there are a lot of good things about a paper and very few sus things, then you're probably fine. Hey, Bobo's here. Bobo, here's a microscopy picture. Bobo is a professional in this field, and they can tell me if I'm wigging out here. So, Bobo, we have here some PI staining for dead cells, and day 4, the background looks a bit high, right? They don't include a lot of detail about their analysis method, and when I increased the brightness, I saw that day 4 is quite a bit of background here. And I would say that this is a little messy, but not that crazy. I don't think that this is like the original image. I'm pretty happy with. It's just that this seems to be an issue of whatever happened on day 4. And I'm not that mad about it. But Bobo, yeah, what do you think, Bobo? I would like to see the analysis, but it isn't weird. Yeah, sometimes you just have like a dirty slide. I say that like it's a little dirty, but I wouldn't be too crazy about it. But if stuff like this keeps on happening in the paper, that's when it becomes an issue. Okay. So we have a treatment salesman. Yeah, I know. Yeah, like sometimes your cells are just dirty. And again, I don't think it's a big deal, but if the rest of the paper is like this, then it is a big deal. But just one individual figure, I think it's fine. Okay, so now that we know that this thing is safe enough to work with, do they actually do the thing that we think that they do? And here I have to introduce a very complicated technique called surface plasmon resonance, which is a lot of jargony sci-fi terminology. And basically how this works is you have some kind of plate that you have a compound attached to. In this case, it is mannose. Oh no, I missed the beginning. YouTube never notified me. Maybe that's for good, because I confused IBS and IBD for like the first hour, which I'm cutting out in the VOD. So maybe it's good that you're here now. If you want the TLDR, where these researchers are making little Katamari balls that fish out E. coli to prevent IBD. And now we are talking about how they know their treatment theoretically works. Yeah, so you have a surface, and that surface is coated with mannose. Let me get out of the way here. Get out of the way here so you can see the... Oh, that's the background. Game capture. All right. So on this plate, you have a surface. This is mannose, and we want to see whether or not we actually have productive binding, right? So so this is the illustration of case A, where you just have the pure sugar, and then you flow over the E. coli binding protein, and then when this binds to the surface, it'll actually change the optical properties of that surface, which you can measure in SPR response units, or SPRRU. Okay, so yeah, it's too hard to explain in full, but I forgot that I had explanatory text here. So yeah, attach a ligand to the surface. In panel A, it's mannose. In panel B, it's our treatment. And in panel C, it's glucose. You can already see, without fully understanding what's going on, sort of some patterns that are emerging, right? So when you get binding, you get an optical response, and then you get a jump in the SPR signal. And these colors down here, this represents an increasing amount of FimH. This should be FimH. I spelled it wrong here. So bigger SPR means more binding, right? So if we take a look at this mannose, I'll explain the mannose, and then you can do the treatment. We attached this surface with the mannose sugar, and this is our positive control, and we flow over the E. coli protein, and the E. coli protein should bind this mannose because that's what it does in nature. And so the more of that FimH we flow over, the bigger the binding response should be. So if we go up here, we see 10 micromolar, we get a little bit of an SPR response, and then all the way down here it's 60 micromolar, we get a huge SPR response relatively, right? When you have multiple different concentrations, this is a shout out for my biochemistry bros. You know that when you do this at multiple concentrations, you can calculate a number called the KD, which is the dissociation constant. This is a measure of binding strength. Lower KD means that you have tighter binding. The TLDR of that is that the KD is a measure of how much you need of a certain substrate to bind up half of a given molecule. And so if you have a smaller KD, you need fewer molecules to bind up half of whatever you're testing. Yeah, so you coat the surface with mannose. This is in figure A. Or the MOS mannose oligomer, which is the multiple mannoses chained together on the microbead. And then glucose. Why do we do glucose? Why do we do glucose? I've heard of surface plasma on resonance in the context of, is it SPR or is it ghost? It's 50-50 either way. That's kind of funny. Gold nanoparticles look purple for the same reason. That's interesting. That's interesting. So the ball grabs E. coli and kidnaps them? Yeah, and then flushes them down the toilet, literally. So this is the principle of surface plasma on resonance. This is an incredibly cool technology used to test whether or not things are binding. So why do we test it with glucose? Why glucose, right? Mannose, this is our positive control. The E. coli protein should bind mannose. This is what we understand. So this shows that our SPR is in fact working, and it gives us a KD for how tightly that E. coli binds to mannose. Then we have our treatment, our MOS, our mannose oligomer. This is the multiple chains of mannose that we're sticking on to the Katamari ball. And then we have glucose. And glucose looks a lot different than the other two. So why do we include a glucose control here? E. coli needs glucose to eat just like all other things. Not for this experiment. This is with purified protein. Purified E. coli protein binding to mannose sugar. Oh, sorry. So if you're late to the stream, you can do exclamation point glucose to get caught up on some of this terminology. So mannose is the sugar on your epithelial cells that E. coli will bind to invade them. Right, E. coli will invade your intestinal cells, but it needs to bind the mannose first to put them in the right location to invade your cells. So Marlon says, is glucose a control? It certainly looks like it, right? So do we expect glucose to bind the mannose receptor on E. coli? Do we expect glucose to bind mannose receptor on E. coli? Yeah, and Gold says, to show it is selective to mannose and not just any sugar. Right? What happens if glucose showed a binding response? What can we say about this experiment? Hypothetically, the mannose receptor should only bind mannose. So if we got a response in the glucose condition, what might that mean for our experiment here? What might that mean for the experiment? What happens if we suddenly find that glucose is binding the mannose receptor that we purified? What might that be indication enough? It's not specific enough? Yeah, technically true, James. I'm thinking more of a practical answer here. What might glucose binding to the mannose receptor indicate when we have a pretty decent mechanistic rationale for believing that glucose should not bind the E. coli mannose? It's not tuned to grab the E. coli and mannose. That is true, but what do we... Oh, let me put it this way. Do we continue with this experiment if glucose shows binding? Then you can't make conclusions that mannose is contributing to the signal. Yes, that's the more experimentally minded interpretation, right? Like, maybe the system that you built, maybe this SPR, maybe this glass slide is just binding everything, right? Maybe it's not, or maybe like something is happening, something is dirty, maybe the optical detector is dirty and it's always showing a signal because glucose should not show a signal, right? So this is, it could indicate something about the specificity of FimH, the receptor. It could indicate something about biology. It could also indicate something about your experimental system, right? And so this glucose control makes me confident that this is real, that this binding is real. Okay, so let's actually interpret the mannose and mannose oligomer results here. So you flow over purified FimH, the protein that E. coli uses to bind mannose, and then you get a set of KDs. So I just said that lower KD means tighter binding. So you see up here that mannose has a certain KD, 2.4 times 10 to the minus 6, and then the mannose oligomer, which is going to be used in our treatment, has a KD of 9.2 times 10 to the minus 10. So what does this indicate about our treatment versus the regular mannose? What do you know about KD, reaction kinetics, binding kinetics, and interpret this data? So this is the data that they give, and we are working together to figure out what this actually means. So it looks like, what we've figured out so far is that mannose and mannose oligomer both bind, glucose does not, right? The bigger SPR signal you get, the more binding you have, and then when you do the math, you can calculate this binding constant. So what do we conclude about our Katamari treatment here? Given what you know about the numbers, what do the numbers indicate about our Katamari ball? The smaller the KD, the tighter the binding, as you've said? Yes, that is true. So if I were to make a conclusion about mannose versus mannose oligomer, what would we say? How are these KD stuff relative to other molecules? Is that a good number or bad number? Like, even if one is better than the other, if it's terrible numbers all around, who cares? That's an excellent question, C.R. Smith. I will answer that in just a bit. But give me your vibe check first. Give me your vibe check first. Give me your vibe check first. Your intuition. What does your intuition say? Well, let me put it this way. How does mannose oligomer compare to mannose? Yeah. Marlon says the oligomer has a KD that is 10,000 times smaller than the mannose, which indicates, you could simplify that to meaning that the mannose oligomer is binding 10,000 times more strongly than the regular mannose, which is a huge, huge improvement, right? We're talking many orders of magnitude. And to get to C.R. Smith's question, well, does this matter? And I'll say that, I wish I had a pharmacology bro in the chat. I believe that productive KDs for drug targets are between 10 to the minus 9 to 10 to the minus 12. So that's indicative of pretty decent binding. 10 to minus 6 is like not that strong in terms of binding strength. It's a little non-specific, but 10 to the minus 10 is pretty good. And what's important for our system is that it is 10,000 times more 10,000 times more sticky for E. coli than the regular mannose that exists on your cells, which is super. I bet when they did this, I bet they were jumping for joy that they managed to show this. This is a very, very cool thing. Wait, if lower numbers are good, aren't the glucose graphs showing really strong binding since it's so low? So it's the KD. So there's a mathematical conversion that converts these curves into this number. I don't have the time nor the willpower to explain Biochem 101 just because I don't fully remember how to calculate KD besides K off over K on. But there's some math that you can do that converts these curves to this number. And for these curves in glucose, you can't even calculate a KD, right? This KD would be some nonsense number. Maybe it's like a negative number. Like if you try to do the same math on this garbage, you're going to get a KD that doesn't make sense. Yeah, so there's no number for glucose because you just can't. This doesn't, when you plug this into the formula, you just get garbage. Twitches and flashbacks, yeah, like KD. I had fun learning it the first time because I thought I was going to be a biochem guy, but I ditched it for neuroscience. Yeah, so 10,000 times tighter binding on the oligomer, which means this is a very, very good candidate for further development. It means that this is very, very sticky and stickier, more importantly, stickier than your cells. So if the Katamari ball is stickier than your intestine, that's a good candidate for rolling these E. coli out of here. We biologists are afraid of numbers. Same. Same. Sorry, I'm a liberal arts major. No need to apologize. I tried to make this digestible for everybody who enters. And you've been contributing quite well to these conversations too, which I'm very happy about because it means I get to share some of this esoteric knowledge with people who are outside of the field, which is always very, very cool. Yeah, sorry. So the glucose KD, you can't even calculate it. Doesn't make any sense too. The formula will break. So what do we conclude? We conclude that the mannose oligomer that we are trying to make a Katamari ball out of, way tighter binding than the mannose. Duplicate slide here, very cool. Yeah, so E. coli protein FimH heavily prefers to bind multiple chains of mannoses over the singular mannose that we have on our cells. This gives us the thumbs up to develop a way to fish E. coli out. Very, very exciting. All right. So, what do we have for selectivity here? So, for this, does the data reflect that we have an improvement in selectivity? So mannose oligomers appear to grab E. coli FimH much stronger than singular mannose. So at least this works for E. coli. Whether or not it works, whether or not the healthy bacteria stay put, that's a different question. And here we have a very, very fun and cool picture. So chat, looking at this image right now, do we believe that these microbeads bind E. coli? So here I have, don't give me your answer right now, but I'll explain what we're seeing here. Here we have a bright field image. This is just using regular visible light of the microbeads. And then here we have the fluorescent E. coli, right? Because you can't actually see E. coli, they're too small. When they are expressing fluorescence, that makes them a lot easier to see. So this is the outline of where these microbeads are, right? And if we zoom into these microbeads, you can see that we have E. coli here in red. And so when you just have the microbeads, no mannose, do we have E. coli binding? These are just the little beads, no mannose attached. How big are these balls, by the way? I'm imagining like a golf ball size, but this is more microscopic. They're visible, but very, very small. There was a picture earlier in the stream. And if you open the paper, which is in the description, you can see it for yourself. You just scroll down until you see a petri dish with tiny little beads. I see very few dots, but I don't really have an idea of how much is normal. Think about where the red is here, right? If this is the boundary of the microbead and these are the E. coli, do you think we're getting sticking? Are these E. coli sticking to the microbead? Right? Because these are the boundaries. This is where the microbead ends, and you see some red dots. Where are the red dots in relationship to the microbead? Where are those red dots? This is an observation question, not an interpretation question. Just give me your observation. Where are the red dots, which represent E. coli? Where are they in relationship to the boundary of the microbeads? The red dots are not on the microbead's boundary. They don't look like they're sticking because they're not on your boundary, not on the bead surface. Exactly. So when you just have a regular microbead without the mannose fishing rod, you don't get binding. Okay, good. What about this picture? What if we have the microbeads that actually have the mannose fishing rod? Same kind of principle. These are the microbeads. These are the bright field images. And then when you turn on the fluorescence, look at what you get. These are the boundaries of the microbead, and look at where the red is now. So, would you say that microbeads coated with mannose oligomer, would you say that that binds E. coli? You can give me your observation. Where is the red in relationship to the boundary, and do you conclude that they bind? Oh, that looks like many more are attached to it now? Yeah, right? This is crazy, right? This is a crazy improvement. That ball is absolutely kidnapping those E. coli. Poor bacteria just trying to live their life. Trying to live their life at the expense of your bowels, unfortunately. They're along the bead surface. Yeah, they're coating that bead. Like, let's compare. This is microbead without any mannose. The E. coli pretty much ignore this. They don't care. But then when you roll this Katamari ball over them with the mannose oligomer that they like to bind so much, you get a bead that is filled with E. coli. Sticky E. coli. All reminds me of lint rollers. Exactly. These are getting lint rolled. Okay, what about the treatment, right? Our mannose bead, our mannose coated bead. What about E. coli that have their FimH broken? This is tricky. Oh, I gave away the answer. I gave away the answer. Unfortunately, I didn't have a educational slide there, but look at what happens when E. coli no longer has a functioning FimH receptor, right? If you break the mannose receptor on E. coli, what do you see? what do? You see the same thing that you saw in slide 1, that the E. coli no longer bind, which tells us, and this is really important for biologists, this paradigm, that this interaction is specific, right? It's not just randomly sticking to the mannose bead. No, this is showing that E. coli is sticking to the mannose covered bead because it is expressing a mannose receptor. And that is elegant, that is beautiful, right? Because one thing that you could conclude for this is that maybe mannose is sticky to E. coli, but E. coli's mannose receptor is not responsible. Maybe there's some other E. coli protein that is making this really sticky. Or maybe something about the manufacturing process is causing this to be sticky in general. What this control does, when you break the E. coli receptor, this shows us that this is very specific, that it has to be this mannose receptor, mannose oligomer interaction that is responsible for this lint rolling, which I think is a very elegant, very nice control. So mannose oligomer binding is FimH dependent. And again, if all of these terms confuse you, you can do exclamation point glossary. What's the thing you always say? Specificity and something else? Yes. Oh, thank you, Pivia. Oh, that's so nice. That, I mean, I recognize a lot of you in chat, but it is nice to hear that I am making some sort of impact with regards to people retaining this information. So specificity and necessity. So E. coli it has to have the FimH receptor. It's necessity and sufficiency. E. coli needs, it is necessary to have the FimH receptor for this interaction to work. So it is specificity and, it is necessity and sufficiency. And so as a biologist, this is necessary, right? This is one of those two concepts, necessary. Do all E. coli have those receptors or just pathogenic strains? I don't know for certain. I believe the paper makes the argument that it is mostly pathogenic E. coli that have these mannose receptors. And in general, if you are a pathogenic bacteria of the gut, you want a handle to grab onto cells with, and I believe that most commensal bacteria don't have the mannose receptor, just because they like to hang out outside of the cells and eat the mucus. Like, why would they need to attach to your cells when they're comfortable eating the mucus that surrounds your cells? Like, they don't need to dive deeper than that. You said earlier that this isn't the actual full E. coli, but just the proteins. So this is the actual E. coli, right? This experiment, this SPR, this is in a dish. This is the purified mannose, purified protein. This is actual E. coli, right? This is actual E. coli. They've been engineered to fluoresce red. So you can tell where they are. Yeah, very, very cool. Very, very cool. Okay, so it's cool that it can do this in a dish, but we should test if this Katamari ball can peel E. coli off of cells. So what do we conclude here? E. coli, which are in red, and since they're so small, you can't actually see them, so the authors have generously highlighted where the E. coli are with these white arrowheads. So you can imagine that the white arrowheads indicates an E. coli that is attached to a cell. So E. coli here in red, aka the white arrowhead, were added to cells with or without microbeads or microbeads plus mannose oligomer. And what can we conclude here? So control, this is a dish with cells plus E. coli and nothing else. This is a dish with cells, E. coli, and the microgels. And then this is our treatment, right? This is E. coli, cells, and our Katamari balls, our mannose covered microgels. So what do we conclude here? What do we say here? Hi Fi, hey Frost, welcome in. We're talking about Katamariing E. coli in hopes of treating IBD. You've entered in a place where we are talking about whether or not this treatment actually works. So the way that this happens conceptually is that these balls that are coated with mannose oligomer that E. coli loves to bind to are hopefully like sticky lint rollers and can tear E. coli off of the cells. And that's what we're trying to figure out if that is true here. So does this work in a dish? Okay, awesome. So do the E. coli not have any way of realizing they've been duped and peel back off the ball, like realizing, hey, there's no food here and leaving? So it's a purely chemical reaction, right? It's a purely chemical force. It is like Velcroing. E. coli can't really fight off the Velcro because it is just physically being picked up out of there, right? And this is another way in which this is preferable to antibiotics, because antibiotics can trigger a stress response in E. coli. They can adapt, they can evolve, they can fight antibiotics. But this is straight up, we're just going to move you. We're going to pick you up and make you someone else's problem, which I think is very elegant. Now, now is natural selection a feature here because the cells aren't literally dying? It's an interesting question. I don't know whether or not this system encourages natural selection. It's beyond my understanding of evolution, because technically, while these cells are not dying, they are no longer able to reproduce in this context, right? Because you've removed them. So I really wonder why. I really wonder if this treatment is suspect to natural selection as well. Using my astute powers of deduction, 3 is indeed less than 14. Exactly. So what do we conclude here? So that's the observation, which is great, right? There are fewer of these E. coli attached to the microbeads that contain our mannose oligomers, and we can reasonably conclude that these mannose oligomer microgels can lint roll the E. coli off of these cells. And it works better than just the microbeads alone, which means that this is a property that is specific to the mannose microgels. Very, very neat. It's not enough that we can do this in a dish, but we have to check an actual animal, right? So when you treat mice by gavage, so if you stick a syringe down their gullets and fill them up with bad E. coli and then give them the treatment, you can actually see that you have a difference in the bacterial populations of the feces, of the ileum, and of the colon. So here you have control. So no treatment, just adding the bacteria. You have the mannose sugar, the mannose oligomer alone. Then you have just the microbead alone. And then you have the mannose oligomer microbead alone. And then you have a positive control here. This is a high affinity mannocide, which apparently they already know works. And then you can see the significance bars. So even though the mannose oligomer and the microgel both look like they decrease the amount of bacteria in the feces, oh, that's interesting. Oh, now I'm confused. I think I have to reread how this experiment was done, because I would expect that the treatment would increase the amount of bacteria in the feces as they're being eliminated. But maybe I don't fully understand how this treatment works. It's kind of interesting. Maybe we'll revisit this figure later here. So this is internal. How does the immune system respond to these foreign bodies in the system? I'm guessing not much, since in the gut the neutrophils don't normally go into. Neutrophils can be in the gut. They can actually squeeze through the epithelium to attack things that are in the mucus layer. So it's not unheard of. And also the immune system is quite active surrounding the gut as well. So they don't know how the immune system reacts to this, because this is brand new stuff. Maybe it's only detecting free bacteria, so the ones attached to the balls don't show up. I think that might be true. I don't remember how they handled or processed these, which is my bad. So I'm not going to talk about this too much because I'm not prepared to. Lamau. All right. So this is an experiment I did think about a bit more, and this is whether or not these treatments actually help an animal recover from something like IBD. So DSS, this is a dextran sugar thing, and this thing, I don't really know how this works, but it induces colitis. So if a mouse ingests this sugar, it damages their insides, right? This is a feature of IBD that they can kind of replicate, but not perfectly. They can kind of replicate this by giving this mouse this toxin. And so we have a system of where you have a mouse, and for the control, you just give them water for 9 days, along with their regular food and stuff. In the colitis experiment, you give them DSS at 2%, and you give them the bad bacteria, and then you give them water to recover after day 7. This is just the microgels. This is the mannose oligomer. The mannose oligomer microgels. This is our full treatment. This is the drug that they eventually want to sell. And then this is their mannose tight binder control. they're positive control. And so you can see that as like, okay, so first this is the control, right? This is the healthy colon. Sorry, I should have had a trigger warning if you're grossed out by organs. But if you're a biologist, I'm sure if you're interested in biology, I'm guessing the vision of organs is not exactly that brutal to you. Yeah. So where was I? Yeah, so we have a couple of different colons here, and we are inspecting them for health. And we can see here, this is the control colon. It has a certain length, which indicates, this is a happy, healthy colon. And then you have the colitis treatment, the sugar poison treatment. You have just the microgels, just the mannose oligomer, the full treatment, and then the positive control. So what do we conclude here from this data while I go back and read the chat? But the mouse immune system isn't going haywire since these were in live animals. We don't necessarily know that. We're only looking at colon length. So maybe the immune system is really upset. We just don't know for certain that it is. If the immune system is really upset, it's not negatively impacting the length of the colon at the very least. Yeah. So as a fungus, what I'm gleaming here about humans from this is that these treatments are trying to bolt a turbocharger to the human immune system. I don't really know if that's accurate. It's more like a Katamari ball or a lint roller, just trying to lint roll these bacteria out of there. I'm more concerned about the smell of the organs. Yeah, not entirely pleasant. What are we measuring here? The intestine length? Yeah, you can see that there's actually a ruler right next to the colon. That is what we are measuring as a proxy for digestive tract health. Longer the closer to the control length, the healthier the mouse is, theoretically. We are hoping for the colon length to be as close to the control as possible, where they were never given colitis to begin with. I have no idea what I'm looking at. So these are a series of colons. These are a series of colons. This is a regular colon. This is a colon that has had IBD given to it, essentially. You can think of it that way. This is a colon that has been given half of the treatment. This is a colon that has been given the other half of the treatment. And then this is a colon that has been given the full treatment. And this is our positive control. That is what we are looking at here. And you can do exclamation point glossary for the definition of these terms. So what are we looking for? What are we looking at here? MOS-MGs and the positive control seem to be the most healthy and longest. Yeah, right? So if we look here, we can see that when you compare to the DSS, this is the colitis IBD mouse, that there is a statistically significant effect of our full treatment. They do have a slightly longer colon, maybe about 5 centimeters as opposed to 4 centimeters. So they regained a whole centimeter. It doesn't really look like the MOS-MGs are necessarily better than this other existing treatment, but I'm guessing this existing M4284 is a bad drug for hypothetically other different reasons, right? This is their positive control. So it's not better than what's out there for this particular thing, but maybe there are other aspects of this new drug that are better. I bet they were a little bummed that their treatment didn't work better than their positive control. Control never got colitis, the rest were all infected, and the rightmost is the best performer of the infected ones. Does it sound accurate? I would say that the rightmost is, when you look at the data, it's about the same as the second rightmost, right? This is our treatment, this is the thing that we are hoping to be the best, and it's just about as good as something that exists out there already. When you look at the data, I agree that when you look at the colons here, that this one looks slightly longer than this one. But yeah, this is just one colon out of like, I don't know, a dozen that they tested, or half a dozen. I don't know. Did they only run this experiment with 6 mice? How do we know that this isn't a fluke? Don't you need more points of data, or at least triple the number of mice? This is a wonderful conversation to have, especially when it comes to mouse studies. So that is not a bad question to ask. But the answer is, when you do mouse studies, especially exploratory ones, you don't need very high ends. In fact, you can't have high ends because, like, this is a conversation I have with Myelin quite often. It is that, in statistics, you are taught that you need at least 30 ends to have a reasonable by the central limit theorem to have a reasonable normal distribution, which means that your statistical analyses are pretty well backed. But in order to do 30 mice, you need a lot of money, a lot of space, a lot of time, and a lot of lost lives, right? A lot of lost lives. These are 6 colons per group, so it's still quite a few mice. But if you imagine, there are 6 treatment groups, and if you wanted 30 mice in each treatment group, that's 180 mice, which is a fortune and a lot of dead animals. And in biology, you kind of have to make peace with the ends are going to be lower than what a statistician or a psychologist might prefer, but it is a trade-off that we make for cost and animal welfare, right? You can do what's known as a power analysis to figure out the number of ends would be reasonable to get a statistically significant effect if there indeed is one, and that informs biologists and gives them a minimum number to aim for, and that minimum number is often less than 30. Overwhelming majority of time less than 30 in an animal. Hopefully that answers your question. So M4284 is the current existing medication. I don't know if it's an existing medication or something they know just binds E. coli. I don't know a whole lot about that compound. MOS-MG is our hopeful medication though. It is our Katamari ball, our sticky ball, our lint roller that rolls E. coli out of the GI tract. Okay. So these MOS-MGs also improve markers of other health. Like, so there's gut health, but you want, there's a colon length, which is a nice way to inspect for colon damage. But we also see that, okay, in orange here, we have the control weight, right? As mice get older, they'll get a little heavier. Mice generally don't lose weight as they age. But if they get sick, like what they have with the DSS in this orange line, their weight will crash because they're having tummy issues. We see that most of these curves are going down here, right? The MGs, the microgels alone, and the MOS alone, half of the treatment is not really sufficient. But we see that the Katamari ball, the MOS MGs, and the M4284, somewhere in the middle, right? Somewhere in the middle of not doing anything and continuing to have their weight crash out and recovering like you would see in the wild type. I don't know what a DAI score is, so I'm just going to ignore this right-hand panel. But weight is something that I can understand, right? When a mouse is sick, it loses weight. And these treatments, they're not perfect. They don't get them back to healthy weight immediately, but they decline less, they decline more slowly than their healthy counterparts. I've had that lecture with some of my colleagues outside of mice. Yeah, it's I think of it primarily as mice because it is one of the most sympathetic angles, right? Because people who aren't really in the field of biology are like, oh, why is the n so low? Sometimes you don't need the n to be that high to have a reasonable power and welfare. Like, I think once people hear, oh, okay, we shouldn't be sacrificing 180 mice for one experiment, they kind of get it, right? They kind of get it. But like, I think that is the more empathetic, like, reasoning to talk with a population at large. But statistically, yeah, you just don't need that high of an n all the time, I would say. Yeah, the colors aren't great, Frost. Mice probably don't live that long to return to full health anyway. I mean, they live for 2 years. It's expensive to keep mice alive for that long anyway. Yeah, if you want to do like a nice thing for a rodent, you can adopt like former lab animals. Not all universities have a program like that, but I used to be at a university that recently ended their rat adoption program where they would allow you to adopt a rat that had an experimental procedure done that would otherwise be euthanized. And that's cool. That's cool. Okay, so how do we know that this is actually working the way we think it's working? So we know that the health starts, their health looks a little better than the regular colitis mice. But how do we know that this is working because we're evicting E. coli? Well, we can check their microbiome diversity. So here are a couple of different metrics. The Shannon index, I'm not going to go too deep into this because I'm not a comp bio or a geneticist, so I don't actually know. I couldn't actually give you an idea of like how significant these effects are. But I say that, like, in most of these cases, the blue bar with the MOS MGs looks closer to the orange bar. So like, sure, like, I don't know. It looks okay to me. I'm not going to say too much about this. Yeah, like these metrics are about diversity. Like how diverse, how many different bacteria species are there? And then there are 3 different ways to calculate it, which I don't really fully understand myself. All right. So what I do like here is when they sequenced the different families of bacteria in the microbiome of these mice, they saw a different population. And I do know from my previous experience with the microbiome that there are two very good populations of bacteria that have been affected here. So you can pause here and just have a look at this a little bit and kind of appreciate what is going on here. So over here in the control, this is the normal, or this combination of mice, the normal flora of the mouse. So you have a chunk of Rickenella. This is like for micro. This is so microbiologists can geek out because I know very little about most of these. Muribacue. I'm not even gonna. These are also hard to say. Muribaculaceae. Don't know. These are for the real microbiologists. But you can see in the control, there's a lot of lactobacillus, and we know, even if you don't know a lot about biology, you maybe have heard, drinking your Yakult or your probiotic drinks, that lactobacillus is a probiotic strain of bacteria. They are good for you, and there's a good chunk of the bacteria diversity that is in the control. And then we have others in green. Bifido in this annoying gray is also quite good. Bifidobacteria. It's a name that I reckon. There are two more that are important, but like to me, Bifido and Lactobacillus are the two big winners because I've come across them more frequently. Yeah. These graphs are frustrating to me. Yeah, these are a little hard to read. They're a little hard to read. MOSMGs are being compared to the positive control. It makes no sense to not include them in the previous set of graphs. I mean, I think it's fine. I think that it's more about showing that this thing works than it is showing that it is better than previous stuff, which can be done at a later point in time, right? Because, yeah, I mean, I think this is a matter of taste. Like, it's completely valid for you to want the positive control to continue to show up here. I think that's totally fine. To me, I'm not too fussed about it because you can refine this technology later in the future. And again, every time you add a group here, you have to sacrifice more mice. So I'm not too upset about that. But yeah, like I think it's reasonable to want that, but I think this is where reasonable people can disagree. I'm not too bothered by it. I'm an ecobiologist, so I study ecosystems, and I use a long-range boomstick for keeping forests clear of invasive species. I'm assuming colitis is pretty bad. It's definitely not pleasant. It's a big decrease in the quality of life for many people. Also, ecobiologist means you're probably very more familiar with measures of mathematical diversity than I am. Very, very cool. At least in my uneducated opinion, it'd make MOSMGs look like the best performer due to how close they are to the healthy control. Yeah, in terms of looking at these bar plots, yeah, I think so. Because if you see in the DS control, this is the, not control, this is the experimental group where you give the mouse colitis. You poison them with this dextran, and you see that whatever group this is, I can't really tell because the colors are so terrible. I think this is the Lacnospiraceae, whatever you want to call it. This really increases, and you see like the lactobacillus, they're almost gone, right? This colitis is devastating for the good bacteria in your gut. Yeah, and I think if my eyes don't betray me, I think the like Bifido are also gone when you give them colitis. Like your microbiome really suffers when IBD has been induced. Then you have maybe some improvement when you give them the microgel, which indicates maybe the microgel itself is doing something, and maybe some improvement when it comes to restoring the mannose when you're using just the mannose oligomer, where you see expansions of like more healthy bacterial populations. But then in the MOS-MGs, you get good recovery too, right? Like even though the lactobacillus recovery looks the same between MG, MOS, and then the full treatment, right? Because this is one half of the treatment, this is one half of the treatment, this is them combined. The lactobacillus seems to recover about the same, but then I think the Bifido up here really comes back strong when you combine these two treatments, which I think is very, very interesting. I think it's very, very neat. Okay, so do the authors make good on their promise? So this is what we have so far. and then we'll fill in the blanks. Oh, it's pretty big here. I think this is, yeah, okay, everything is still on screen. So does the data reflect better limited bioavailability? Well, it seems to work in this model of DSS-induced colitis. It's pretty nice. As Frost had pointed out, they don't really finish up the experiment, including the positive control, which maybe they did for good reasons. I don't really know. But at least in this model, it seems to work. Whether it works better than existing stuff, we don't really know. Does it take care of this gap of repeat administration? So most of their experiments don't really last for all that long, and IBD is like a chronic thing. So this is kind of a limitation in the fact that like colitis, DSS-induced colitis is like a one and done thing. You can poison them one time and then you throw their microbiome into terror, you damage the gut lining, and then you can see how long it takes to fix that. But it might not be the same molecular etiology as real IBD. Yeah, so these mice didn't have real OBS. Oh my God, IBD. So who knows? And then did they address stability and safety? So the individual components from a hypothetical perspective seem harmless, and the mice seemed grossly okay, like they didn't die. That's great. And the drug didn't have an effect on the immortalized colorectal cells, so they also looked grossly okay. So who knows if they're toxic in some other way, maybe they impede function of certain things, but they didn't die, which means that this is probably a pretty decent candidate to try and test further. But for an exploratory paper where they are first demoing this, I'm pretty happy with it. Okay. So how sure are we that this is how this drug works? How sure are we? Right, this is the proposed mechanism. Mannose oligomer microbeads reduce E. coli binding to cells in vitro. Mannose oligomer microbeads improve colon physiology after DSS-induced colitis. Mice treated with Mannose oligomer microbes show improved marker of health after induced colitis. Mannose oligomer microbeads reduce bad bacteria and increase good bacteria. This is their data. This is a pretty bare bones interpretation, right? I'm just reporting what they found. I'm not interpreting it at all. But how sure are we that the way that these mice are getting better is that we are doing a lint roller and rolling the E. coli out of the gut? How sure are we? Is this enough proof to show that this is definitely how this mechanism works? So the next step is like primate studies. I would maybe not go right away to a primate. I think there's a lot more that you can do in a mouse before you subject a primate to this kind of experiment, because mice are very expensive. But a primate is like next level expensive. It's not even in the same ballpark how expensive it is to have a primate in your lab versus a whole bunch of mice. I don't want to be the poor intern cleaning the mice cages with the ones with OBS. I feel bad for the vet techs. It's not always the graduate students who have to do it. Yeah, so how sure are we that the lint roller is actually removing the bad E. coli, and that's what's making the bacteria, the mice feel better? How confident are we that this is definitely how this works? And you can maybe tell by my wording where I stand on this issue. We don't have the complete story, but we have an indication. A peer review or repeated studies can help us get a better understanding. I would say that it's not so much peer review or repeated studies. I would say it's more like not peer review, but replication in separate contexts, right? Like this is a good start, but there's still a lot more that you can do to show that this is worth spending millions of dollars turning into a real drug. Yeah, so I think that's probably what you meant, Frost, but I just wanted to kind of touch up the language a little bit. All right. So it's a good start for certain, I would say. But hypothetically, what can we test just to push experimental thinking? If we wanted to be more sure that the way that this drug works is by rolling E. coli out of the digestive system, what could we do to improve the odds or refine the odds that this is actually how this thing works? Because it could work in other ways. Like, what other ways could this be working? right? It's kind of open-minded, or it kind of shows like there's still a lot more work to be done. So 2 questions. How can we further test this to make sure that this is in fact the mechanism by which this drug works? And 2, what other explanations for this drug working could potentially be true given what we know about the data? From the singular paper, it seems not terrible at this. We need replication in a different lab to be more sure, something replication crisis. While it would be nice to do it in another lab, it's probably not going to be done in another lab because this is their baby, right? This drug design is their baby, so they are going to be the ones to try and sell this to a company at some point. Yeah, so the way that the replication crisis works is a little different for drug design, right? The replication crisis for drug design is a big pharma company buys your idea and then they can't get your drug to work because biology is complicated, and that has nothing to do, that doesn't really have a lot to do with the flaws of how research is done. It's just sometimes you make an investment and it doesn't pay off. They could be preferentially breeding on the ball. That's all I could really think about for other ways it could work. I want to be very specific here. How do we know it's specifically E. coli that it's affecting, as an example, right? There are a lot of unanswered questions here. We know that this ball binds E. coli, but what if it's binding other things? What if not only it gets rid of the bad E. coli, but gets rid of other bad things? Maybe it doesn't get rid of bad E. coli in the gut. Maybe it gets rid of other bacteria in the gut. There are a lot of dimensions of uncertainty that can be refined with more and more experimentation, although I do think this is a fantastic start. So here's my suggestion. Maybe you can inoculate mice with either normal E. coli or E. coli with a FimH mutation, the FimH being the mannose receptor, induce colitis in both groups, treat both groups with mannose microbeads or regular microbeads, or a control of, vehicle, saline, PBS, whatever. And then if it is specifically E. coli competition, what should we see in this experiment? I would be concerned about how these beads would be impacting signaling pathways in the gut that revolve around these cell surface sugars. This is an interesting question. Like, I don't actually know how this bead interacts with mucus, for example. Yeah, that's awesome. No, that's a really good concern. Because you can imagine if this bead is sinking into the mucus and clogging up the mucus and hanging around for a really long time, that could be kind of bad for cell-cell signaling. It could be bad. That's smart. That's a really smart limitation that I never thought of, Gold. What background do you have, Gold? That's like a very good biology answer. There's like DNA testing that can be done. I know they do mass DNA reading for bacterium to tell what exact species are there. Yeah, for certain. But we already did that. We already have a population level understanding of the diversity of the different bacteria post-treatment. So what should we see with my hypothetical experiment? So I'm doing the necessity/sufficiency thing. I have normal E. coli, normal pathogenic E. coli, that is. You inoculate them, you give them colitis. And then another group I have, one with the FimH mutation, broken E. coli. So if E. coli has a broken mannose receptor, do you expect that this Katamari ball treatment should work? I'm an unemployed biomed major. Nice. Hopefully that changes soon. We will have employment. I'm also unemployed, so we have YouTube is side money, and I am also in that same spot. So there's solidarity in there. What was I saying? So if there is no mannose receptor on the E. coli, do you expect the Katamari ball to have an effect? If it has a broken mannose receptor, it shouldn't grab on the ball. Exactly. So this is an experiment that I came up with. So if E. coli mannose competition is responsible for the microbead success, mutant FimH E. coli will not be effective and colitis will not improve. However, is this a good experiment? This is an experiment that I came up with, and I like this experiment theoretically, but now's your chance to tell me that this experiment is actually bad. Because it is. This experiment is not great. They should not do this experiment. I think hypothetically it could work, but there is one fatal flaw in this hypothetical experiment that would make this probably not work. And can anybody identify why this experiment is probably not great and why stuff like this is so hard to study? Why is this experiment likely not going to work? Because I would say, hypothetically, if you could get this experiment to work, this would be very, very good proof that the way that this ball works is almost for certain because it tears off E. coli from the gut. But I'm saying this experiment doesn't work. And why might that be? Why might that be? Give you a second to think about that. Will broken E. coli not cause colitis? Well, you're inducing colitis, but you're hovering around the correct answer here. Does mutant FimH also make colitis in the same way that non-mutant ones do? Yeah, so Sierra Smith and Piffy, you are both hovering around why I think this experiment is bad. It also seems like it can go south fast because colitis is not exactly the most compliant disease. Yeah, it's hard to make it for sure, but with the tools that we're given, I think that this is hypothetically good, but in reality, pretty bad. So I'll give you a hint. What's the problem with an E. coli pathogen that cannot stick to your epithelial cells? What's the problem there? Almost by definition. How is that problematic for our experimental design? Why might that be a bad experiment? Why might that not generate useful information, I should say? Pathogenic E. coli sticks to the epithelial cells. E. coli with a broken mannose receptor. And would it select for E. coli that attack the gut through a different mechanism? Maybe, but that's not where I'm getting at. Controlling for any mouse's bacteria, gut bacteria from before feels difficult. That is true, but you can do fancy things like microbiome-free mice that don't have bacteria in their gut, and then gavage. Can I repeat the question? Yes. So my suggested experiment to really nail down the mechanism behind this Katamari ball is to inoculate mice with either normal E. coli or E. coli with their mannose receptors broken. And then if you treat both groups with your Katamari ball treatment, you should expect the E. coli with a broken mannose receptor to not improve. Like, the drug should not work if the E. coli mannose receptor is broken. But I'm saying that this is also a terrible experiment. Yeah. Chat is getting it now. With a hint, chat is getting it now. Would E. coli with broken mannose receptors even be pathogenic? Yeah. If it can't stick, it can't do its bloody job. Yeah. It can't be causing the problem if it can't even stick to the epithelial cells. I would say that, yeah, all the combination of all of this and the fact that you just, like, if they can't attach to the cells, they might just get flushed out. You might not have productive colonization of the gut with an E. coli receptor mutation, which is maybe why they never tried this experiment, because it's hard to interpret. It's not exactly a good experiment, even though if hypothetically this were to work, it would be pretty definite proof that this is in fact the mechanism, at least in this experimental system of DSS-induced colitis. So yeah, good job, chat. With a little bit of a tip, I think you all got the answer right. Like, this kind of shows you how elegant your experimental design needs to be, because this is an idea I just kind of crapped out, and it's not tremendously clever, and it doesn't even work. But hopefully I've illustrated how fun science can be when you're thinking about how to tackle these experimental problems, because the limitations of your system will force you to be creative. And I like that about science. So even though hypothetically this sounds good on paper, it's probably pretty stupid. At least that's what I think. Yeah, so I think this is pretty bad. Shows you how complicated it can be to nail down a mechanism with precision. So when people say, again, I'm so one-guyed by that dumb comment from the schizophrenia paper, but like, if you see people complaining about mechanism, now you know it is really hard. It's really hard to nail down a mechanism with precision. You can get hints about a mechanism most of the time. You can have plausible models that you will continue to refine, but science is slow and progressive, and oftentimes you never get a perfect picture, but you get a picture good enough that you can actually develop some treatments, which is, I think, very important to keep in mind. Want to hear a biology joke? Sure. So here are the conclusions. Oh, good question, Sierra Smith. Does it matter the exact mechanism if it does its job properly? Like, I don't care why it works and that it does. A lot of medicine is like that, and I think it's fine. I crashed out in my schizophrenia stream because there was a stupid comment on a hacker forum about thinking that the DSM, the Diagnostic Statistical Manual for the Treatment of Psychiatric Disorders, is useless because we don't understand the mechanisms of psychiatric disease. And like, that's the dumbest thing I've ever heard in my life. You don't need a full, complete understanding of disease mechanisms to treat them. You just need to make sure that they are safe and that they do their job. The more you learn about a disease mechanism, the better you can refine the drug to reduce side effects. But yeah, Sarah Smith, I think that's a good and healthy attitude to have towards medicine. As long as it's safe and it does its job, you should probably take it, but you should also hope that we understand it better so that you can make the drug even better than it was before. What's an immune cell's favorite firearm? I don't know. Excited to hear the response. Some rage baiter out there is ready to type, wait, it isn't useless? Yeah. I'm making it too easy to get one guide. But yeah, sometimes I like stopped when I was like having a productive day of editing, and I had to like do an emergency journal club essentially to rage at that one commenter. Okay, so conclusions. Instead of trying to kill populations of bacteria, which can have off-target effects, right? Antibiotics are not exactly very specific, and you want to keep the good bacteria and kill the bad bacteria. This group proposes to gently relocate bacteria by luring them with a mannose oligomer to mimic our own epithelial cells. The initial safety profile was very rudimentary, as expected. This is an exploratory study, a new technology, but it seemed to help recover after DSS-induced colitis, right? It seemed to help the mice gain weight or stop losing weight at such a fast rate, I would say. It helped them with having a healthier colon, and it seemed to remove E. coli from adhering to cells, at least in a dish, right? The real biological environment is, of course, more complicated, but in a dish, it seemed to work as intended. And it also seemed to restore healthy gut microbiota after DSS-induced colitis. We saw recovery of lactobacillus populations and bifidobacteria, and 2 other populations of bacteria that I don't really remember. And then the final conclusion is all hail to Katamari. I don't know if Katamari really influenced these authors, but I certainly see the parallels, and I think are very, very cool. Well, I think it clicked in my head. You couldn't tell the difference between not working because of broken E. coli or no effect because of the beads. Exactly. That's a perfect way of putting that, Piffy. The results would be not interpretable because the broken E. coli are bad at doing their E. coli job anyway. All right. So that is all I have. What did y'all think of the paper? What did y'all think? What did y'all think about this Katamari drug design paper? And do you like drug design papers? I usually don't cover this kind of thing, right? Usually I do basic biology because I think nature is the best engineer of them all, but sometimes it's occasionally nice to appreciate the ingenuity that humans can come up with as well. So I'm wondering what y'all think about me doing more of these drug designy papers. Nucleotide pistol? I don't actually know what that's a reference to. I'm sorry. I don't get it. I don't get it. If he says very clever stuff, I think so too. It's clever because it's, you know, it's something that anybody can think of. Bad bacteria in the gut, why don't you just lint roll them on out of there? And then it just takes some expertise to figure out exactly how to lint roll it out of there, but the idea is very simple. Wondering if nature already does something like this. Maybe. I don't know about the gut to know for sure. It took me until someone asked you to repeat the question to get on the right direction. I can't believe I derailed myself so hard that I lost the plot of the question. I mean, it's kind of a complicated idea, and I didn't spend that much time on this journal club. I think this was maybe a 3-hour investment, reading and putting together the slides. I've definitely spent longer, and I've been more careful with how I word things. So maybe it was my fault for not being too clear to begin with. I loved following through with this. Please do a lot more of this. Yeah, I think so. I think as long as you can find good candidates for drug design topics, I think this kind of thing is very, very fun. I'm still waiting for the nanomachines. They were promised to me since the 2000s in the documentary. Yes, the documentary Metal Gear Solid. Well, I actually do think that there are nanomachine medicines out there that are catalytic. I don't want to say that for certain because I don't remember exactly what they are, so I don't want to say that for certain, but I do remember seeing nanomachines being used in medicine. Little hinges, little protein hinges that stick two things together, and can bring host proteins and enzymes in proximity to one another to help a crappy enzyme do its job. Things like that, I think, will be explored much more fully in medicine in the future. But yeah, like if you want to look into nanomachines in a medicinal context, I believe there is a hinge protein, a small nanomachine that bends and can bring things closer together, which I thought was neat. The closest we have to nanomachines in the modern day is supermolecular chemistry. I think they do call that thing a nanomachine, though. Maybe that's maybe someone with more chemistry or nanotech technology can correct me, but I do think they technically call that one a nanomachine. With my very limited medical knowledge, I hope these microballs will balance my humor so I don't need to do bloodletting. We should hope. How do the gels get into the mice gut? Are they able to survive oral ingestion in the stomach? I don't remember. It's probably in the paper, which is linked in the description. They gavaged with E. coli. They could have definitely gavaged the beads, but I don't remember. It's a good question, though. Good question that I probably should have read, but again, I did not spend a lot of time on this journal club. Given the fact that I confused IBS with IBD for the first hour. Still have to edit that part out. All right. With that being said, I am hungry. I am going to introduce nutrients to my own digestive system. And I think I'm going to call that stream, y'all. Thank you all so much for hanging out here with me today. And do I have anything to plug? Let me check the VTheorem calendar. Let me check the VTheorem calendar real quick to check if there's anything to plug from other people. VTheorem calendar. Oh, we have Z's Twitch debut at 1 p.m. on Friday, 1 p.m. EST on Friday. So if you are more likely to watch on Twitch, you can look forward to that. And now I'm always so bad at like redirecting streams because the Twitch, the streaming stuff on YouTube is like so bad. But let's see if there's anybody I would be interested in redirecting to. Looks like that is not happening now, but if you want to check out friend of the channel, Charis Karis Euphranor, a philosopher VTuber who I have a lot of fun talking to and hanging out in chat, I will redirect to him, and you can go check him out when he streams. I believe he's streaming at 4 p.m. today. That's in another 4 hours from now, but I guess this video will redirect to him anyway for some reason, which... Okay, YouTube, thank you. Very cool. When am I making a plush? I will make a plush when I make enough money on the YouTube channel to actually save up to buy. Okay, so here's a little behind the scenes for the merch thing, right? For those of you who are interested in. I actually recently bought a lot of my own merch. I have a merch store. It's not public yet. I'm buying the contents now to make sure that the merch is of sufficient quality that I would feel good about selling it to all of you. But the problem with plushies is that you have to buy like 300 of them in advance. And there's no way I can afford to buy 300 plushies right now. But, you know, hopefully if this channel makes money, I will be able to do that because I really, really want to. If you want to support the channel, of course, you can do membership, you can super chat, etc., etc. And in a couple of weeks, hopefully, if the quality of the merch is good, you will be able to buy your own Fi merch, which I am very excited about. I really, really love the hoodie design that's coming out. I cannot wait. I've been talking to Toxel all day. I'm like, I can't wait to get my merch. I can't wait to get my merch. We'll have like stickers and magnets and a hoodie and a hat. They look really good, and I'm very, very excited to share them with you. It is very, very exciting. Actually, quality checking, not very capitalist to me. I mean, I would feel really bad because like I don't expect people to buy my merch in the way that like, oh, you know, I'll just put something out there and people will like it. I want to really make sure that if you buy it, you should be happy with the product. It should perform the function you expect it to perform, we'll just put it that way. And like I really want to make sure that you end up with a quality product. A 5 plushie would be so cute. It is an option, but until there is excess money, until there's a surplus that I can invest, it'll have to wait a bit longer. Just put something out there and people will like it. I see if you've read about the Bethesda gaming approach to products. When you're too big to fail, you can get away with that. But, I think I'm always going to have, one, I'm a little guy, right? I'm a little neutrophil. But two, I think I always have little guy mentality, right? I think in order for me to be happy, I can never think of myself as the big guy. And making sure that the quality is actually good is a very little guy thing to do, because as many of my stream viewers know, I'm a very high ego. I think of myself as the protagonist-centered kind of person, which I'm aware of. I do think of myself as like wanting to be an anime protagonist, right? Wanting to be a science idol. I have high dreams, high expectations for myself. But in order to do that, I need to make sure that I am happy with what I am doing. And I'm hoping that the samples of merch that I am going to get soon, hopefully, are of good enough quality to share with all of you. I definitely had the same experience with Audacity merch. Turns out our logo is really unfriendly to DFX. I'm not sure what that is, but I'll write in DTG. I don't know what those acronyms mean. Always happy to catch these when I can. Thank you so much for dropping by, Frost. Everyone is their own protagonist. I think so, but I think I like actively believe it. Like I'm actively aware that I think of myself as a protagonist just because I think it's more fun that way. Like I think it's fun to lean on thinking of myself as a protagonist. I think version of a protagonist is okay to have. Yeah, I think so. Like you don't have to view yourself as a protagonist the same way I do. You could argue maybe not even healthy to do so to begin with, but like because my ego is so massive, I like have to be. Here's the thing: my ego is massive because I think I have been checked so hard repeatedly in my life that I have now gained an understanding of how big my ego is and how best to keep it in check, right? But like the protagonist energy, the big ego protagonist energy, I think is why I think like I don't want to speak this into existence, knock on wood, but I don't think I'll ever like burn out on making this kind of content for you all because I just enjoy it so much. And I feel like if I make content that I enjoy, like I'll say this, I'm incapable of making content I don't enjoy, right? I think a lot of YouTubers burn out because they feel beholden to the algorithm. But as I've said in my membership videos, like I think my ego is so big that I think that whatever I think is good, you will think is good too. And the ego is big enough to believe that there are enough of you out there that I have to find that can support me in making that kind of content. That's kind of what I mean by big ego. Like, I don't feel insecure in the face of the algorithm yet because I think my taste is good and I think the content that I make is good. But the thing is, you kind of have to believe that to make content, right? If you think the content you make is bad and you continue to think that way and you never believe that it's good, then it becomes very hard to do. So, like, I say that my ego is big, but it's more like I really believe in myself is maybe a better way to do it. And me saying my ego is big is more like self-defacing, like making a joke out of it, which maybe isn't so appropriate. Because some people might actually believe that I'm an egomaniac, when in reality it's just I, for now, really believe in what I do. Yeah. Well, how many times do you really think about how other human beings have their own stream of consciousness and wants and dreams and are a protagonist to themselves, and you're a stranger to them? I think that there are people in my life that I view as having protagonist energy. And like that, it's like that's not to say that everyone else has NPC energy, right? But there are some people that I know personally that make you want to root for them. Whereas that's not necessarily true for everybody. That's what I mean by protagonist energy. They have a goal that you want to cheer them on for. Now, like I have, friends, and of course I wish them well. But like, you kind of know what I mean? Like not everybody lives in a way where they sweep you up into whatever stuff they're on, which is fine because, you know, maybe they're working on something amazing, but they don't have a huge ego like I do and want to like share that. Maybe they're working on something in secret. Maybe they're just working in their day-to-day life to live a nice and comfortable existence, right? Like this is not like, this is a judgment-free thing, but I think people who have protagonist energy make you want to cheer for them. And I think that is externally protagonist facing, right? It is the appearance of protagonist. So everyone can think of themselves as protagonists. And of course, most people, you don't really think of them being protagonists because you're worried about your own stuff. But I think there are a couple of people I know who I'm like, I want you to win. Succeed is not even the right word because I want all of my friends and everyone I know who I like to succeed. But there are certain people who will have protagonist energy that I want to win. I want them to win whatever they're trying to compete for, is what I should say. I hope that makes sense and doesn't come across as too like crazy, right? Because I'm not saying that like, again, it's not a moral judgment. It's an observation that some people's protagonist energy ends up sweeping you along with them, and you can't help but get caught up in whatever cause they're dedicated to. Always choosing to make your own decisions and leverage your own agency gives you the power to be the protagonist of your own life. Yeah, like people who, and that like describes the correlates of what I think is like protagonist energy. People who have a vision and then they just do it. You know, in absence of pressure or insecurity, they just figure out a way to make the thing that they want happen. And I admire that. Not universally, because sometimes that's bad, right? I can name a few very famous people who maybe have too much power to affect reality. Some say an outsized ability to make their impact on reality. And I don't view them as protagonists. But the people who you generally like and agree with, you know, morally, ethically, I think it makes me want to cheer for them. I think I've never seen any content that you've made that doesn't align with what I want to see on the internet. Toby Fox made the kind of content he wants to see so that people who like it would see it, even though there's people who think his humor is cringe. Oh, mentioning Toby Fox in the same like thought as me is incredibly endearing, like incredibly nice. Because I do think Toby is like similar like protagonist energy, right? Like I wish he would finish Deltarune before I died, right? Because one of my biggest fears is that I will be involved in some horrible motor accident and I will never know how Deltarune ends. But his is another like protagonist energy where he had a vision, he made it happen, and it's good. It's good vision. Now, notice I say protagonist and not hero because I don't think we should put people on a pedestal, but it's okay to like view them as like people who can who make something that you cheer for, I'll say. Ideally, this is what the algorithm wants to model. It's AI and its way to predict what a viewer would have wanted to watch next anyway. Yeah, like I like the algorithm in the sense where it is exactly showing you what you are interested in. I just really hope that I can do a good enough job in front of enough people to make it a sustainable thing for a long-term career. Loving your own content is a key of some of the best creators I've seen. Yeah, and I think the ego joke is that I love my own content enough so that like if I were to ever to make changes that would make me not love my own content, I wouldn't be making it anymore. It's impossible. Like I can't. In some ways, I'm kind of a child in that way. I can't chase the algorithm. Like I've tried, right? I have documents, scripts that are algorithm chasing, but I never end up making them because the process is so disgusting that I just stopped trying. Yeah, I missed all of biology, didn't I? Yeah, but you know, there's a VOD that I have to edit real quick. Gotta head out. Thank you for hanging out, Seed. All right. Heroes feels like it's universal. Protagonist feels nicer since it adds the nuance of something that can shift with perspective. Yeah. I think that's really the thing. Like, a hero is like unambiguously the good guy in the story. Protagonist, I think, is more the person that you root for, and it has layers of nuance. And yeah, I would like to be a protagonist because I internally view myself as a protagonist actively in that kind of way. Like I want to be a science idol, right? In that kind of way where that's not something that people have done before. But I think it would be cool, right? I think it'd be cool to be that for people who want to engage in science in this specific kind of way. Yeah. All right. I'm getting very, very hungry. Thank you all for hanging out with me. Go watch Z on Friday on her Twitch debut. And later today at 4 p.m. EST in 4 hours, you can go watch my buddy, Mr. Euphenor, the philosopher VTuber. And maybe I'll see you there if I get bored of editing and want to hang out. All right. Goodbye, y'all. Goodbye. See you at some point. Bye-bye. Bye-bye.